ফোন কেটে দিচ্ছে, ধরছে না। ঠিক আছে, চেষ্টা করবো। তাহলে আমার কাছে ফোন তো দেখতে হবে, তুমি টাইমটা লিখে দুটো লিখে রাখবে না আমার কাছে ফোন।
ওখানে ক্লিক করুন।
সুভাষিস বাবু জয়েন করেছেন?
হ্যাঁ, জয়েন করেছেন। চন্দনাদি,
এটা স্টপ না হলে তো।
ওনাকে আপনারা পার্সোনালি ফোন করুন, কারণ উনি বোধহয় শুনতে পাচ্ছেন না।
আমি করছি।
সময় তো হয়ে গেছে।
বলেছি, দেখা যাক।
থ্যাংক ইউ সীতা ম্যাডাম। ওহো, ওকে।
এবার হয়েছে মনে হচ্ছে।
মৌমিতা সামন্তকে ইয়ে কর, এন্টার করতে দে।
এলেই হবে, আসেনি আমার কাছে।
আমাকে বললো ডিনাই হয়েছে। মৌমিতা
Okay.
সামন্ত @gmail.com দেখ আছে।
ওকে ওকে, ঠিক আছে।
আচ্ছা, ইউটিউবে লাইভ আচ্ছা, লাইভ ব্রডকাস্টিং কিন্তু স্টার্ট হয়ে গেছে।
ওকে, তো Convener madam, principal sir, should I start?
Yeah, you please proceed.
Okay. A very good afternoon to one and all. On behalf of Postgraduate Department of Zoology and IQAC TDB College, Raniganj, I welcome you all to this two-day international webinar on confronting the impact of COVID-19 pandemic. Since we have received lots of responses, it's not possible to accommodate all the participants in the Google Meet platform. Therefore, we have arranged for a live broadcasting through our YouTube channel. The link for which has already been shared. The outbreak of COVID-19 pandemic has created a global health catastrophe that made a deep impact on the ways we perceive our world and everyday lives. This unprecedented pandemic is engulfing the world into a deep darkness and is rapidly spreading across nations. Countries responded with lockdowns, forcing people to stay locked at their homes, silencing the busiest places with waves of fear and death. We are moving closer to the 10th month of pandemic, and there seems to be no relief from the highly infectious COVID-19 pandemic which is ravaging the world. The crisis in India in a worse manner than we could have imagined. Hence, our response to the pandemic has to be more specific and effective to get rid of pandemic from the root level. Even when a vaccine reaches us, we would need prioritization and regulation in place to ensure that a COVID-19 vaccine reaches all corners of the nation. As of now, more than 165 vaccines are currently in different stages of preclinical and clinical trials, and more than 33 of them has reached the critical stage of human trials. The good news is that even if the vaccine is not completely effective, it will be still effective in controlling the spread of the pandemic. When people go back to business as usual, more and more people will get infected with the disease and develop immunity against the virus. However, herd immunity may come at a price which will be beyond our comprehension. Sooner or later, we will understand the power of togetherness. Locked in our homes, fighting with an unknown, unseen enemy with no definite cure in sight, we are down with depression and various other psychological problems. This webinar attempts to address the need of finding a ray of hope to combat the various physical and psychological problems faced by us while leading a cocooned life of social distancing. With this short note, now I would like to request our respected Principal Sir, Dr. Ashish Kumar De, to deliver the welcome address. Sir.
Hello. Hello.
Yes, sir. You are audible, sir.
Good afternoon to one and all. It is indeed a proud moment for my college as the postgraduate department of zoology of our college has organized the international webinar on confronting the impact of COVID-19 pandemic in collaboration with the IQAC of our college. I take pride to witness the excellence of an academic institution which has traversed a long, glorious journey of 60 years of academic excellence with its head held high. This institution offers graduation courses in 28 subjects and postgraduate courses in 7 subjects. It is one of the largest colleges in West Bengal, but webinars like this enriches its academic quality in many ways. At the very outset, at first I welcome all you. I have a great proud privilege to welcome Professor Dr. Sadhan Chakraborty, Honorable Vice Chancellor, Kazi Nazrul University, Asansol, for giving his consent to act as a chief guest of this international webinar, in spite of his busy academic schedule. It's my great pleasure to welcome Dr. Sumedha Rai, postdoctoral research scientist, Department of Biomolecular Medicine, Faculty of Medicine and Health Science, Kent University, Belgium. A warm welcome to Dr. Rajkumar Koiri, Assistant Professor, Department of Zoology, School of Biological Sciences, Dr. Hori Singh Gaur Central University, Madhya Pradesh, India. I am also a great proud to welcome Dr. Moumita Samanto, Senior Scientist and Team Lead, Dr. Reddy's Laboratories, Hyderabad. I take the opportunity to welcome Dr. Rupesh Kumar Srivastava, Assistant Professor, Department of Biotechnology, All India Institute of Medical Science. A warm welcome also to Dr. Animesh Banerjee, Postdoc Fellow, IMCB, A-Star, Singapore. Actually, the present moment, we are living on the edge of the two sides. On the first side, it is well known that we have exploited the environment and Mother Nature in a way that the hand that robs the prey is in severe tremor. The climate is in despair, which poses a dagger in disguise to our existence. So it is now the need of the hour to care and nurture the nature back to its glory. Not only the misuse of environment and ecology, we are also living on the edge of COVID-19. It has seen that the coronavirus COVID-19 is the defining global health crisis of our time and the greatest challenge we have faced since World War II. COVID-19 is much more than a health crisis, and humanity needs leadership, solidarity, to defeat this pandemic. With this spirit, the zoology department, our postgraduate zoology department, has organized this international webinar on confronting the impact of COVID-19 pandemic to discuss the different impacts, challenges, and also, I think, some of the psychological effects of the pandemic on society. I hope and believe that this webinar will succeed with all its objectives. I'm sure this will be a rewarding venture for all of us present here. Before I end, I again welcome all of you, all the teachers, all the research scholars, all the experts, all are welcome. Thanking you all. Thanks.
Thank you, sir. Thank you for your encouraging words. We will now begin our technical forum. Our first speaker is Dr. Shumedha Roy. Before going into the formal introduction, I would like to mention that my association with Dr. Roy dates back to the year 2011, when I had my acquaintance with her as a very efficient, amicable person at the Department of Zoology at the University of Burdwan. And indeed, I could request her to be our resource person for this international webinar. She obtained her doctoral degree from Bose Institute, and her research work involved animal physiology. Dr. Roy was engaged as an associate professor in the Department of Zoology at the University of Burdwan. Her research interests include ecotoxicology, molecular toxicology, and cell biology. She has actively worked with Drosophila melanogaster to perform the potential effect of different toxins. She is also an editorial member and reviewer of several international journals and has supervised several PhD scholars. Dr. Shumedha Roy is a member of many international affiliations. She has been recognized and certified as outstanding reviewer by the Royal Society of Chemistry for Toxicology Research in 2016. She has authored many research articles, books related to ecotoxicology, molecular toxicology, and cell biology. She was also engaged in active research in the French National Institute of Health and Medical Research, Lille, France. Dr. Shumedha Roy is at present working as a postdoctoral research scientist in the Department of Molecular Medicine at Ghent University, Belgium. She is currently exploring the different mutations which lead to rare recessive neurodegenerative diseases, demonstrating ataxia, dementia, chorea, and hypogonadotrophic hypogonadism. Today, Dr. Shumedha Roy would enlighten us about the different basic yet vital aspects of the pandemic through her talk entitled Panic Amidst the Pandemic. Please may I have the honor to hand over this forum to Dr. Shumedha Roy for sharing her views with us. Please, madam.
Thank you. It is my pleasure to be a part of this forum. Actually, I'm grateful to Raniganj TDB College, who has given me this opportunity to share my views. Though I'm not a virologist and not a person who is working on COVID-19 at present, but I would be very happy to share some of the informations that I gathered in the last few months regarding the COVID-19 and its association with the nervous system. Means what is the effect of COVID-19 in the nervous system. And but before that, I must say that Ongona, it was too much. Means you just made everything very difficult for me because I thank you, a lot of thanks for all the good words that you told about me, but they were really very, very big thing. I don't know whether I can live up to that expectation, but I'll try to share my views with you in the next few minutes. So do I start sharing my screen?
Yes, ma'am, please, you can.
Yes, please, ma'am.
Is it, can you see the thing?
Not yet, ma'am. It is just on the right side of turn on Captain.
Yeah,
Isn't-
I saw that. Okay, let me open the tool then. Why is it?
It is loading, ma'am. Give some seconds to load it, okay?
Is it visible? Why is it not?
Ma'am, is it showing you are presenting the whole screen like that? Any command?
Yeah, I want to share the whole entire screen. And
just
I,
click on present now, then it will come as you are sharing the whole screen.
yeah, that
You are
I did
sharing-
actually.
Okay.
And now can you see?
You have to just click on the screen which is showing. Then first click that, and now it is.
Yeah.
Yes.
Now it is.
It is
Yes,
coming.
ma'am, now it's showing. Yeah, it's okay now.
Yeah. Fine. Okay. So I will be talking about panic amidst pandemic, and here I just, as I told you that I would not be telling you anything related to viral infection and how the pathogenicity of this virus is actually causing the respiratory distress and all. But I will be talking about panic amidst pandemic related to the neurological implications of COVID-19. So from the topic, the name of the title of the lecture, that is panic and pandemic. Let me start. What is panic and what do we mean by pandemic? Panic is just a sudden feeling of great fear that cannot be controlled and that actually prevents us from thinking very clearly. And when we talk about pandemic is the worldwide spread of a new virus, which has been defined by WHO in 2010. So panic is a situation which is really difficult for our mind because our uncontrolled fear takes over us and we cannot think properly, we cannot act properly the way that we want to. And really, now is the situation where we can see that we are in a position where we are getting panic stricken because of this pandemic, COVID-19 pandemic. And to start my talk, I would just remember a few words from William Shakespeare's Hamlet, where he says that diseases desperate grown by desperate measure are relieved or not at all. Seriously, we are in a situation when we are in desperate need of anything that can relieve us from this disease. So no or not at all, I'm not going for not at all. We are looking forward for a brighter tomorrow. So we are in desperate need of anything that will actually help us to get out of this disease. So coming to this point where I must say that how we reached this 10 months of COVID-19 pandemic, how this was started. How this started from December 2019, where a coronavirus was detected in Wuhan in association with life-threatening severe respiratory distress. And WHO named that novel coronavirus as SARS-CoV-2 because of its similarities with SARS-CoV, means the other coronavirus that were previously known to be associated with another respiratory distress which originated in 2003. Because of the clinical and the virological similarities, they were both termed as SARS-CoV and SARS-CoV-2. So from that point of view, the SARS-CoV-19 infection started spreading, and just like a bush fire, it took over the whole world, and WHO was forced to declare it as a pandemic on 11th March 2020 in the press release that, yeah, COVID-19 is a pandemic. And the viral, there much has been talked about how this virus attacks the humankind cells and how the virus takes over the infection. I will not go into that detail, but the thing is that regarding the virus, I must say that it possesses RNA as the genetic material, and along with the RNA, it has some proteins. There are four main structural proteins involved in the process of infection. They are the spike proteins, the membrane protein, the envelope protein, and the nucleocapsid. So here we can see that there are different kinds of proteins that are involved in the process, and among this, the spike protein plays a very important and vital, relevant part in the process of infection. But I'll not go into detail with how the spike protein is involved, but let us know that the spike protein, which is present on the outer surface of the virus, does play a role in the process of the pathogenicity. So coming to the point that this coronavirus is one of the major viruses that targets, that we all know that it is the first respiratory distress that starts. So whenever we are talking about coronavirus, COVID-19, the first thing comes into our mind is that it is a respiratory, means it is a virus that targets the respiratory system. But here comes the point that I want to share with you, that this virus has a neuro-invasive capability. So it can attack the nervous system as well. Our central nervous system can be attacked by the virus. So how far and how well, that I will be sharing a few views, some of the works that has been dealt with in the last few months of this pandemic, that I will be sharing with you. And this spike protein, which is common for SARS, the previous SARS and the CoV and this SARS-CoV-2, because these spike proteins use the same angiotensin converting enzyme receptor type 2. This receptor is present in the human cell, and that is taken up by this virus, means that these receptors are actually recognized by the spike proteins, and the spike proteins make contact with the virus, with the human ACE2 receptors, and that is how it it starts the pathogenicity. Now, the third point is that the ACE2 receptor here is widely distributed in the body, present in the lungs, in the oral or the nasal mucosa, in the bone marrow, in the spleen, skin, heart, arteries, kidneys, adipose tissues, everywhere, and also in the brain. That is what I want to point to, the fact that this ACE2 receptor, which is responsible for binding the viral spike proteins, are also present in the brain. So this ACE2 receptor is also expressed in the central nervous system, and that is in the brain, in the different parts of the brain, that is predominantly in the thalamic nuclei, in the cerebellum, and in the inferior olivary nuclei of the brain. So these locations of the presence of these receptors in the brain may be very relevant, and it is actually responsible for the cause of coronavirus encephalitis. It may be linked to the encephalitis. So now coming to the point that increasing evidences are being available that suggests that this coronavirus causes the neurological deficit. So it is very important that when we are talking about the coronavirus and the nervous system, so how far the neurological activities or how far the neurological function is being affected, that we want to know. So it has been seen by different literatures that SARS-CoV-2 causes neurological deficit, and it is in substantial proportion of the affected patients that show this neurological deficit. Now, while these symptoms arise actually during the course of infection, but we don't know what is the long-term or the medium exposure or the consequences that the brain is going to face from the infection, this COVID-19 infection. So actually we are not, we are still we don't have enough evidence, or we don't have enough studies that can give us the real picture of the long term or the medium term exposure of this virus in the brain. So it is still in a very primary situation that we are discussing the thing. So now there are there are four possible ways that the pathogenic mechanism is brought about. So directly the COVID-19 can cause the viral encephalitis. It can also cause the systemic inflammation. It can cause peripheral organ dysfunction, and it can be the cause of cerebrovascular changes. So now, but most of the time, the patients that actually shows the neurological deficits, they do it by the combination of all these four. More or less, a combination of all these four things are seen. So now coming to the fact that in a group of scientists from Spain, they actually forwarded a very speculative review where they talked about the possible medium and the possible long-term neurological consequences arising from SARS-CoV infections, COVID-19 infections. And they are of the they actually emphasize the fact that neurodegenerative and neuropsychiatric diseases are mainly of neuroinflammatory origin. So here I want to say that when our brain cells are getting infected by COVID-19, then there is a chance that there is the switching on of neuroinflammation. There is inflammatory signal that actually cause the neuroinflammation in case of the central nervous system, and that neuroinflammation is really alarming because this can be brought about by the cytokine storm from the virus infection. So here I must go stepwise. Actually, there is a problem. I cannot see anybody's face now since I have shared the whole screen. So I just want to see, I'm just going out and I want to maybe share the thing. Am I audible?
Yes, ma'am.
So I want to share the screen, but I want to see people because I cannot see everybody for that reason. How to make people, I can present the screen as a window, no?
Yes, ma'am. As a window, you can do it.
Okay. So the whole of the screen is now visible.
Can you see now? We can see you actually.
Yeah, that is a problem. I cannot see everybody of you, and that is where I am just. It's difficult for me because I want to see your faces. So, okay, I cannot see, but it's okay. Let's continue. I don't want to waste time. Regarding this, I must say that when SARS-CoV-2, that is the COVID-19 virus that infects a human cell, then it initiates the cytokine storm. Actually, this cytokine storm is known to be responsible for neuroinflammatory signal in the central nervous system. So this, since I have already told that the COVID-19, that is SARS-CoV-2 virus, has a neuroinvasive capability, so it is actually getting inside the neurons. It can get inside the neurons and switch on the cytokine storm. When this cytokine storm is switched on, it might switch on the neuroinflammatory signal. And this neuroinflammatory signal might be leading to the onset and progression of neurodegenerative and neuropsychiatric disease of neuroinflammatory origin. So this idea is speculative, but it cannot be ruled out because it is really alarming. I will come to different studies in the next few slides where I will say how this is very relevant and what can happen if there is a long-term infection of this COVID-19. But first of all, I must say what is a cytokine storm. Cytokine storm, also known as the hypercytokinemia, is a physiological reaction in human and other animals where the innate immune system is actually, it causes an uncontrolled excessive release of the pro-inflammatory signal. So it actually causes the release of excess amount of cytokine. These cytokines are nothing but they are the chemicals which are responsible for initiating inflammation. So they actually push the cell towards inflammatory signal. And normally the cytokines are part of the body's immune response, and it is the part immune response for any infection. But if sudden release of large quantity of cytokine can lead to multi-organ failure and can lead to death. So that is how cytokine storm is taking over in case of this COVID-19 infection. So how this cytokine storm is relevant and how it can be alarming, that is what I'm going to present in the next few slides. So now coming to the next part, that how this cytokine storm is related to the nervous system. The cytokine storm has a devastating effect on the respiratory system that we have already known. We have already seen that several people are dying across the whole world. The death toll is continuously rising. So we can understand that cytokine storm is associated with the respiratory system, but how it is relevant for the nervous system. So it has already been reported by Poyati et al., another group of scientists who were working with COVID-19 patients, and they came across a woman who had fever, cough, and altered mental status. So she was not behaving properly, so her behavior was a little bit different from normal. And when she was observed, she presented necrotizing hemorrhagic lesions and encephalopathy. So it was more or less necrotizing hemorrhagic encephalopathy which was found in the patient's brain. And there they actually reported that this actually was caused by the intracranial cytokine storm and the blood-brain barrier disruption. So the woman who had COVID-19 infection, so other than fever and cough, she just demonstrated a change in the mental status, and that was because of the disruption of the blood-brain barrier and the cytokine storm that is going on in the brain, and that caused the necrotizing hemorrhagic encephalopathy. So from there, the scientists were interested to seek that how far this pro-inflammatory response is associated with other neurological diseases. So here, the chronic neuroinflammation that I'm talking about is a neuroinflammation because of the pro-inflammatory signal from the cytokine, excess cytokine. So this chronic neuroinflammation has been reported to be associated with multiple neurodegenerative diseases. So it is not new. It has already been worked out for several years that neurodegenerative diseases like multiple sclerosis, Parkinson's disease, Alzheimer's disease, Huntington's disease, amyotrophic lateral sclerosis, all these common neurodegenerative diseases have been seen to be associated with chronic neuroinflammation. So in case of just taking examples of few to make it a little bit clear, in case of Alzheimer's disease, we know that the beta amyloid proteins, means there are plaques which are deposited in the brain, and that is because there is a pro-inflammatory cytokine release where the interleukin-1 and interleukin-6, they go high, and they causes the inhibition of the phagocytosis of beta amyloids. So that is how the amyloid beta increases, the load increases and gets deposited in the brain. So that is how pro-inflammatory signal is associated with Alzheimer pathology. And in patients with Parkinson's disease, it has been seen that when post-mortem was done, then high level of TNF, IL, and interferons have been detected in the brain. So we can thus say that common neurodegenerative diseases are seen to be associated with pro-inflammatory signal. So what will happen if the COVID-19 also has the pro-inflammatory signal going on, increasing the inflammatory signal in the brain? So here I will just point to a fact that neuroinflammation has been found to play a major role in the pathogenesis and in the psychiatric diseases. So it's not only the structural brain structure that is going to alter, the brain function, the neurotransmitter metabolism can also be affected. So that is what we are going to, I'm going to say that neuroinflammation can induce changes in the structure, function in the brain, and also in the cognition and behavior. So the person can start behaving differently. That person cannot have the same understanding that the person used to have when that person is having chronic or a long-term exposure to COVID-19. That might be one of the reasons. So now coming to another fact that we should also be aware of the possibility of long-term infection that have been shown to be associated with cognitive and psychiatric disorders in children and adolescents. So here I must say that any person who is having the infection should also be assessed for the cognition and the psychiatric disorder also. If there is cognition deficit or there is psychiatric disorder persisting in the patient after recovery from COVID-19, that is also a thing that should be taken care of. Now, if I talk about the direct neuroinvasive potential, I have already told that the virus can attack the cell, means the neurons, means it can directly enter the brain. So the ACE2 receptors that I have talked about is also present in the brain and are highly expressed in the epithelial cells of the respiratory and digestive system, and also in the neuronal and the glial cells in the central nervous system. And this shows that I must say, interestingly, it has been shown that in mice, the virus SARS-CoV, means not SARS-CoV-2, but the previous one, which is very similar to the COVID-19, this SARS-CoV has been shown to induce neuronal death in mice, directly invading the central nervous system. So that is one point that is very important. And other than this, I just want to say that SARS-CoV-2 may also enter the central nervous system via hematogenous route. So it can actually travel through the blood and can enter the central nervous system. But interestingly, this encephalopathy is seen with severe illness, and whatever patients that have been monitored in the last few months, they only don't exhibit the encephalopathy. They exhibit encephalopathy with accompanied metabolic abnormalities. There are several other symptoms that are also very apparent in these patients, and so the encephalopathy might be a result of the cumulative effect of all the metabolic abnormalities going on for this arising from the cytokine storm. And more or less, patients have demonstrated less direct invasion of the virus into the brain. So it is more indirect effect of COVID-19, maybe the cytokine storm, maybe the main cause of the encephalopathy, not the direct presence of the virus in the brain. That has been shown by some of the scientists. And recently in another review, it was said that no treatment until today, no treatment has demonstrated high level of evidence of proven efficacy. So no treatment has been so effective against this kind of condition. Now, coming to another thing is that when SARS-CoV-2 it remains in the central nervous system for medium to long term. That is what I was telling, that we don't know, because it is only 10 months of COVID-2 SARS-CoV-2 that we have come across. We haven't come across 2 years of COVID-2 exposure. So we are yet not in a position to say that what will be the result of, on the central nervous system when a person gets infected, has a long-term exposure to the virus. So viral latency is another important point which should be studied. And interestingly, or maybe I will say alarmingly, it is not about SARS-CoV-2, but a very related coronavirus has shown that it can stay in the central nervous system over an year after the inoculation in the mouse model. So it is really very, very much alarming that this virus is a very close relative of SARS-CoV-2, and it has been shown that this virus can be detected in the mouse model of coronavirus in encephalitis after 1 year of inoculation. So viral latency is another point that should be addressed and that should be studied more carefully. So now coming to another point that it started off in China, Wuhan, we have already known, and in most of the patients that have been dealt with in China, they have reported that about 36.4% of the patients demonstrated neurological disorder. So it is another important feature of the disease that there is associated neurological disorder in quite a substantial percentage of patients. And headache is a very common symptom of COVID-19, which is more or less observed in nearly 40% of the patients that have been diagnosed with COVID-19. And here I must say another report which actually presented the MRI. When the brain MRI was done, the thalamic region and the temporal lobes of the brain, they showed up with abnormal changes in the brain. That these MRI images I will be just presenting in a few slides from now. And I want to say that the markers of the coagulation means other than the virus entering the cell, other than the cytokine storm which is initiated by the viral infection, there is another chance that I told that the virus can make its way through the hematogenous routes. So here there is another point, that is stroke can be initiated. That is cerebral stroke can also be another cause of death for the patients, because the markers of coagulations are seen to be increased after the COVID-19 infection, and there is intravascular coagulation that has been seen. And in one of the patients, the MRI image that I'm going to just show a little, maybe 1 or 2 slides ahead, there you can see that the brain structure is totally altered, and any layman like us can also understand what are the changes that occurred in the brain. And interestingly, a very important study in Seattle was done, where only 8%, the real difficulty of the thing is that whenever we are studying the patients, we are observing, it's a small cohort. So it's very difficult to get statistical analysis being done and statistically relevant data being published. It is very difficult because it is only few months of COVID-19. So, but in case of Seattle, they actually did a study where they showed among the number of patients that were admitted in the hospital, nearly 8% of the patients, they demonstrated stroke. So it is another point which should not be ruled out. So acute necrotizing encephalopathy is one point, which is which might be originating from the cytokine storm, and the other is the stroke infection, the stroke caused by the increase in the coagulation markers might be another cause of the changes in the brain. Now, the most common thing that we have come across in the past few months is anosmia and ageusia. So anosmia and ageusia are conditions where there is a loss of sense of smell and the loss of sense of taste. And this arises just because of the presence of the receptors. Means we know that the upper respiratory infection is common, where we lose the sense. It's a very common thing whenever we have a cold, normal cold, not a COVID-19 cold. Whenever we suffer from cold, sometimes we cannot smell, so we lose the power of smell, and that has been attributed to the damage of the olfactory epithelial cells that are caused by any virus, the viral upper respiratory tract viruses. And that is also true for COVID-19. A high expression of the ACE2 receptors in the oral mucosa is the cause of the loss of the sense of taste. So when we are talking about loss of sense of smell or loss of sense of taste, it is just because the presence of the ACE2 receptor in the mucosa, oral and nasal mucosa, which is actually responsible, which is actually affected by the virus directly, and that causes the thing. But one thing here, again, I must stress that acute disseminated encephalomyelitis, which is a condition, more neurological condition, which has been seen in patients or in people. These people are found to be having viral illness in the past. So any effect on the means any person who has viral illness, that may trigger an autoimmune response which affects the central nervous system or the peripheral nervous system. So a person who has already have the viral infection are more prone towards development of this neurological deficiencies or the central and peripheral nervous system abnormalities. So that is where the importance is that we should not take it as only a respiratory virus, but we should try to address the neurological implications as well in the far stretched days. So it is common that it was seen in a 66-year-old, another case study, where acute thoracic myelitis at the height of illness with SARS-CoV-2 infection was seen. So a person who had, there were no classic cases of ADEM in association with SARS-CoV-2, but this person, after having thoracic, after having developed high COVID infection and respiratory distress, it started demonstrating, the person, the 66-year-old man started demonstrating thoracic myelitis. So the association cannot be ruled out completely. Now coming to a paper of this month, which came out this month only, that if we actually see the approximate timeline for positive diagnostic tests, the clinical presentation and the pathogenesis in the COVID-19 associated neurological diseases, there I must point to this red blocks, where we can see that the cough, fever, tiredness, myalgia, headache, they actually are seen from very early days. Means on the x-axis, it is the onset of systemic or respiratory symptom of COVID-19. So it is from 0 day towards more and more until 14th day or the 2 weeks of infection. That is the normal quarantine period. And so the demonstration of cough, fever, tiredness might be demonstrated from 2nd to 3rd day of infection. Means the respiratory symptoms come up, but what we cannot understand there, the encephalitis, the myelitis, the Guillain-Barré syndrome, these are also taking place at the same time and maybe much before that. So Guillain-Barré syndrome is a condition where the neurons or the nerve cells are being attacked by my own immune system. So where a tingling sensation in the numbness and the tingling sensation in the peripheral nerves can be detected. That is the first sign. And so these symptoms can initiate prior to having cough, fever, and tiredness also. So these conditions should not be ruled out, and we should not neglect this aspect. That is what I want to present before you. Now, coming to the images that I was talking about. So these are images of the brain from 6 different individuals, where I think it is very apparent that when the MRI, the T2 flare MRI, that is flare means fluid attenuated inversion recovery, where basically the CSF, the cerebrospinal fluid, appears dark, and here the imaging has been done in such a way where very clearly we can understand that in the right lateral ventricle there is a ventriculitis, a change in the structure, an accumulation, uh, a different appearance in the right lateral ventricle is very clear. Now, coming to the second patient, where the hyperintense signal in the right medial temporal lobe of the hippocampus is very clear, and here also it is actually this condition is a sign of encephalitis. So there is hippocampal atrophy, the hippocampus is reduced, and there is a sign of encephalitis which is prominent from this image. And in the third case, where the hyperintensity with the bilateral medial temporal lobes and the thalamus is very clear, we can see the change in the brain. And in the fourth patient also there is hemorrhage which is indicated. And in the fifth person we can see the ischemic lesion is there, so there is a constriction or there is ischemia, there is less of supply in the brain, and it is involved in the left lobe, occipital lobe. And in the sixth patient that I was talking about is the patient who suffered stroke. So it is a middle-aged man. I might say that it is middle-aged because 64 is not a very aged individual. So that person, the gyrus of the precentral gyrus of the brain in this 64-year man gets disoriented or deteriorated neurologically, and he was diagnosed with a stroke, acute stroke. So these are the conditions which are, these MRIs make it clear that the conditions are not very easy. So now I must say, I will just point out one fact that I think we should be aware of, because in the society there are several patients who are actually suffering from neurodegenerative diseases, and these patients should be under regular observation, regular consultation. But due to this long quarantine period, or due to this long lockdown period across the world, these patients are suffering a lot, because neurological patients, they are mostly progressive degeneration that is occurring. And when this long 10 months of no consultation, no physicians face to face, so they are actually suffering a lot. So a reprogram consortium, they came up with an idea that we should not actually stop the treatment of the neurological patients, rather we should come up and carry out the consultations, their regular networking. Only we should be a little more cautious about hand hygiene, about masks and all. But it is never accepted that the patient should not be treated, because the caregivers who are at home looking after the patients, they can understand that the situations are deteriorating. And many people who in this last 10 months have developed some symptoms of neurodegeneration, cannot go out to the doctor, and they are showing deterioration, and these people are at a risk. So if they get infected with the virus, then the conditions would be much more severe than any other normal healthy person. So this is where we should think a little bit that treatment should not be stalled because of this long lockdown and this long quarantine pandemic conditions. And we should also take care of another important aspect, is that patients who are elderly patients living in the nursing homes or elderly care facilities, because in this part of the world where I am now staying, here there are elderly people who live in nursing homes. Nursing homes are actually kind of old age home of our country. So these people who are already aged, who are having low immune system is a little bit weaker than the healthy normal individuals, these care old elderly people should be given, or should be, maybe we should take care of them by thinking of having vaccination. Means we should think of pneumonia vaccine, we should think of influenza vaccinations for these elderly people. I think that might be another solution. This idea has also been forwarded in a paper a while back. And now I know that I captioned my lecture as Panic Amidst Pandemic. So I have told many things about the neurological implications of COVID-19. I know that is really panicking thing. We must be panic stricken. We are afraid, but I think this is not the right time to be afraid of. Rather, we can choose to not to be afraid, and we should think that we can channelize our energy, our anxious energy, into the direction, into the right direction. So I came across a paper where they actually discussed. I like this cartoon very much, because there's a spectrum of panic consequences due to COVID-19 pandemic, and here is the positive and the negative. So let's make a pledge that we will actually channelize our anxious energy, the fear and this energy that we are spending towards being afraid. We will channelize that energy towards the positive, to get informed, to get to reach out to other people, to just extend our hand to help out to others. And there I will just ask my young friends if some students are there in the audience who are young, and who still have the ability to go out and reach out to others and extend their helping hand, to stay connected, to be positive, to give a thought that we will not ponder over negative thoughts. We will not be wasting much of the energy in searching informations from the internet, not so good sites. Means I'm saying that not so authentic sites of the internet, that is actually increasing the anxiety in the young people. And let's step aside from getting from being misinformed to being really informed, and be prepared with our body. Let's exercise a little bit, let's be compassionate with others, and after all, we should, if things are going out of hand, let's take a breath and relax. And if that at all is not helping, then please go out for professional advice. So let us all take a pledge that we will be on this side with a little bit of optimism, with a little bit of health consciousness and information, being more informed. Let's not go towards this sadness, paranoia, annihilation, and this nihilism and annihilation and all this. Let's go to the right. Now, this is what I wanted to share with my young friends, and these are the literature and websites that I've consulted while making up this lecture. But the last slide, I want to say that taking the words from Honorable Principal Sir of Raniganj TDB College, I'll say yes, we have exploited nature. We, the humankind, have exploited, maybe we have over-exploited nature, and we have gone into the animal habitats, we have endangered many species, we have exploited nature for our benefit, for so that human being come up as triumphant. But this is the high time. I liked one word from Roy T. Bennett, the positive thinker, who says it is better to find your courage to make changes when you can choose to, rather than being forced to. If you do not make change, then change will make you. So, this is what I believe. This is the right time to make change. We should stop invading the animal habitats. We should stop invading the biodiversity hotspots. I will again quote a famous Bengali author, Bonnera Bone Sundor, Shishura Matri Krore. So let the forest remain there, let the animals who live in the forest remain in the forest. We don't need to go up to them, because we don't know what is the how they are acting as reservoirs for many of the unknown viruses, and we can get exposed to them. So let's reduce the disruption of the pristine forest, and let us focus to a discipline which is the planetary health, which actually takes care of the links between the humankind and the ecosystem, and the way in which we can keep the balance. And after all, with all this different information on how we are afraid, on how everything is going wrong now, but I believe that together we can overcome. So we shall overcome, and every cloud has a silver lining. Waiting for the bright sun to come. Thank you, everybody. Okay.
Thank you, ma'am. Thank you for your such a nice, insightful and informative talk, which was more of a revelation about the conditions we are facing in our daily life, about the difficult times we are facing during the pandemic. And I hope that our viewers who are watching with us through the YouTube platform will also acknowledge that your talk would help us to confront the present situation in a better way. I also request our honorable speakers to please stay with us, because we have a question hour forum at the end of all the talks, so that we can accommodate the maximum number of questions. So with this, I would like to go forward to the technical session. And one more small announcement for our viewers: please post your questions or comments in the comment section of our YouTube, so that those can be dealt with during the question hour forum. Now, our next speaker is Dr. Rajkumar Koiri, whom I take pride to introduce, as he's our very own alumnus, and we are very overwhelmed to get him as a resource person. I proudly say that Rajkumar Da has studied in our very own Department of Zoology, Triveni Devi Bhalotia College. He has obtained his MSc and PhD degree from Banaras Hindu University. He is an assistant professor at Dr. Hari Singh Gaur Central University, Sagar. He has published several scientific papers on cancer biology, therapeutics, pharmacology, and toxicology of different cyanobacterial toxins. Dr. Koiri's current area of research is to study the metabolic adaptation for survival occurring in the cancer cells. His research topics include targeting phosphodiesterase to enhance drug delivery and overcome drug resistance at tumor site in carcinoma, He also works on molecular mechanism and structured function analysis of cyanobacterial toxins. Dr. Rajkumar Koiri is the recipient of the DST SERB Young Scientist Award. He is the life member of Indian Society of Cell Biology. He is serving as managing editor of Frontiers in Bioscience, guest editor, and editorial board member and reviewer of numerous scientific journals. Today, Dr. Koiri will talk about the different immunological aspects of the coronavirus through his talk entitled Immune Response to the Viral Infection. Please may I have the pride privilege to hand over the session to Dr. Rajkumar Koiri for his views. Thank you. Please, sir, you can continue.
Thanks. Dr. Angana, am I audible to you? Dr. Angana, am I audible to you?
Yes, sir. Yes, sir, it's fine. Yeah.
Thanks, Dr. Angana Ghoshal, for a nice presentation. I would like to thank the organizers, particularly Dr. Ashish Kumar De, Principal TDB College, Dr. Arun Kumar Mukherjee, Dr. Rupalendu Ghosh, Organizing Secretary, my old colleague, old friend, Dr. Tapan Pahal, Chandana Khan Madam, and other members of the organizing committee for providing me a platform of TDB College from where I completed my graduation almost around some 2 decades back. And this stay in TDB College has played a vital role in molding my present career. And I'm going to share my views on immune response to viral infection. And for me, basically, it's a homecoming. I have tried to keep the talk as simple as possible, keeping in mind that we have undergraduate students also. Basically, I will be talking about immune response to viral infection in general. I will not be focusing on COVID-19, as there are a number of talks on COVID-19. As suggested by the organizers, if required, maybe I will go to bilingual, or if required, I can go to trilingual also. I Bangla bolte pari, so no issue. Okay, so I'm starting my talk. Just one minute. Is my screen visible?
No, not yet.
1 minute. It's asking for grant permission also, but it's not asking.
You can do one thing. You can mail me your PowerPoint, then I can share it if you wish. From my.
I'm just signing out, and again I will join just with.
Okay, 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 okay.
must grant permission in order to screen share. Please ensure whether any permission has to be from the organizer side, because it is asking me can't share your screen. You must grant permission in order to screen share.
No, there is no need of permission, sir.
Because on my screen I am getting a message. You can't share your screen. You must grant permission in order to share a screen share.
It is. Is it asking permission for presenting the screen?
No, it's saying you must grant permission in order to screen share, and I am getting the only option is that to dismiss. I'm not getting any other option. Normally, whenever we present the screen directly, because I think meeting has been organized by you people. Please take if some permission has to be given by you.
Rajkumar da, then you can send me the presentation. Means as far as I know, my permission is not required.
This type of things never used to come.
Yeah. This never happens actually. So you just mail me your document.
So no issue. I'll mail you.
Yeah. Then I'll share it from my side. Yeah.
What I was concerned was that every time we'll have to change the screen, because if you'll present, then I will not be able to change the screen as and when required.
Yes, then that will be a problem. You have to say change the screen, and we have to change it.
Every slide. My screen is visible to you. Organizers, Dr. Ramana.
Yes. Yes, it is visible now, sir.
Yes, but some kind of noise is coming. Okay, your screen is visible, but some kind of it's visible, but from the background, some kind of noise was coming. A shrill noise was coming from the background. Some kind of noise is coming. Okay, your screen is visible, but some kind of
Now it's clear. now just tell me now. Now the audio quality is good, or is still some echo is coming around?
Now I think it's okay.
Okay. So
Yeah, now it's fine. Now it's fine.
yeah.
Yeah.
Okay. So I'll be talking about immune response to viral infections, and this is a very simple talk. I will not be talking about much about COVID-19. I will be talking about what happens during a viral infection, how our immune system how our immune system participates in encountering of viral infections. Okay, so let us just talk one by one. So the first thing is, how does an infectious virus cause a disease? So if we'll go through this diagram, you will find that it's a generalized diagram of a virus, in which you'll find that there is an envelope surrounding the virus. On the envelope, you will find there are envelope proteins, and here you will find inside a nucleocapsid we have got the viral genome. So in order to cause an infection, the first thing that a virus has to do is to that first it should infect a host, and after infecting, the most important point is that virus must reproduce or replicate itself. So in order to reproduce or replicate itself, the virus first it attaches itself to a host cell. After having attached to a host cell, it injects its genetic material. Okay, that genetic material means this is the viral genome. So it must inject its genetic material inside the host cell. During uncoating, replication, and assembly, the viral DNA or RNA incorporate itself into the host cell's genetic material. And
Excuse me, sir. One
yeah,
second. Sorry for interrupting you. Actually, your presentation is not being seen.
okay, 1 minute. Is my presentation visible, Dr. Angana?
Yes, yeah, now it's visible. Your presentation is also visible. Your audio is also clear now. Yeah,
Okay.
you can carry on. Yeah, thank you.
So in this presentation, we'll discussing about immune response to viral infection. So the first question is, how does an infectious virus cause a disease? So here you will see this is a generalized diagram of a virus, where you will find there is an envelope. On the envelope you will find there are different types of envelope proteins. For example, if we if we say take the example of HIV, then on the surface of HIV you find certain glycoproteins like GP120 and all, and based on those GP120 and all, we can perform antibody interaction, and we make use of those principle of antigen antibody interaction while performing ELISA or Western blotting in order to detect the presence of a virum in the serum of a patient. So, in case of each and every virus, you'll find there is an envelope. On the surface of the envelope, you'll find there are envelope proteins. And here you will find that inside the nucleocapsid, you'll find that this is the viral genome. So, in order to cause an infection, the in order to cause an infection, the best part in case of virus is they create a self-sustaining system. Self-sustaining means once it incorporates its genetic material inside the host cell, as the host cell replicates its DNA, the virus genetic material of the virus itself will be replicated. So this is how a virus replicates. So in order to reproduce, the virus first attach itself to a host cell. It injects its genetic material, and during uncoating and replication and assembly, the viral DNA or RNA incorporates itself into the host cell genetic material and induces it to replicate the viral genome. And this is how it ensures that its progeny are transmitted to another host. In order to transmit its progeny, first it needs to increase the number. So this is how it creates a self-sustaining system by which it ensures that its progeny are transmitted to another host. And then we'll talk about the different mechanism of transmission. So the different mechanism of transmission includes that can be via aerosol, it can be fecal oral, it can be vector-borne, or it can be close personal contact. So, aerosol is transmitted via inhalation of droplets. For example, in case of the common cold virus, rhinoviruses, or in case of adenoviruses. It can also follow the fecal oral route. For example, in case of astroviruses, calciviruses, and these viruses are responsible for causing acute gastroenteritis. Or some of the mechanism of transmission are also dependent on certain vectors. For example, in arthropods such as mosquitoes, ticks, fleas, for example, this type of mechanism is being adopted by arboviruses family. Then it can be via close personal contact, especially exchange of bodily fluids during sex or via blood transfusion, for example, during herpes viruses. This is a generalized slide showing that in our body we have two types of defense mechanism. One is non-specific defense mechanism, the another one is specific defense mechanism. So whenever we talk about non-specific defense mechanism, non-specific defense mechanism includes skin present on our body surfaces, mucous membrane, secretion of skin and mucous membranes. Then we have another line of defense that are where phagocytic white blood cells participate. We have antimicrobial proteins. We have inflammatory response. And coming to the immune system, that is considered as the third line of defense, we have got lymphocytes, we have got antibodies. Okay. So, in order to cause a infection, the first prerequisite for a virus is to it has to gain entry into the host cell. Okay. So, before getting into the entry into the host cell, it has to bypass certain barriers like skin. So basically, skin is a dead cells that there. So basically, the outer surface of the skin is basically a dead cell, and the advantage of it being in dead cells is that it cannot support virus replication. So in order to gain entry into the host, most viruses which infect via skin requires a breach in the physical integrity of this affected area. There has to be some cut, or maybe there has to be certain abrasion, so that the virus can gain entry. Okay. So many viruses employ vectors like ticks, mosquitoes, vampire bats to breach this barrier. Now, next is our respiratory tract. In contrast to skin, the respiratory tract. So, if you can remember well, जब भी हम लोग को सर्दी खांसी होती है, we have cold. You people can see that the flow of mucus increases. So inside the nasal passage, we have got mucosal surfaces which produce mucosa. So in contrast to skin, the respiratory tract and all other mucosal surfaces possess sophisticated immune defense mechanism. For example, there is presence of ciliated epithelium, there is a presence of mucus secretion, and one more important thing is lower temperature. Remember, in order to survive, in order to survive for a virus, even a difference of few degrees will not is not suitable for the survival of viruses. So all each and every viruses have got an optimum temperature. So in case of respiratory tract, you'll find in the respiratory tract we have got lower temperatures, and these are the things that viruses must overcome in order to survive. Then we have got gastrointestinal tract. Gastrointestinal tract has got a very hostile environment. For example, in our stomach we have got gastric acid, we have got bile salt. So whatever goes inside to our mouth, first of all, it has to pass through the gastric acid and bile salt, and many viruses they fail to survive this hostile environment of gastric acid or bile salt. Then we have got genitourinary tract. Genitourinary tract is relatively less hostile than above, but it is at the same time it is less frequently exposed to extravenous virus. Then we have got conjunctiva in the eyes, which is an exposed type but and relatively unprotected. So, so these are the common sites of virus entry: eyes, mouth, respiratory tract, alimentary canal, skin, urogenital tract, and there is one thing more important. It's not that the day we are exposed to a virus we become disease. There is an incubation period, and this incubation period may vary from a number of days, and it may go up to a number of years in certain viruses. So if you will go through this slide, you will find that in case of influenza, the incubation period of the viral infection is around 1 to 2 days. In case of common cold, it is 1 to 3 days. In case of dengue, it's around 5 to 8 days. In case of poliomyelitis, it's 5 to 20 days. In case of measles, it's 9 to 12 days. And in case of certain diseases like hepatitis, it's around 50 to 40 days. In hepatitis B, it is around 50 to 150 days. In case of papilloma virus, it's around 50 to 150 days. And in case of HIV, remember it's 1 to 10 years. इसका मतलब if any person has, अगर किसी पर्सन को अगर HIV का इन्फेक्शन हुआ है मे बी, तो उसको HIV के डिजीज, रिमेंबर there are two terms, one is AIDS and the another one is HIV. So HIV doesn't miss AIDS. HIV stands for the virus. AIDS stands for the immunodeficiency syndrome. So a person can be infected by HIV, and maybe that person is not going to manifest any symptom for the coming 5, 6, 7, 8, or 9, 10 years. His symptom of AIDS will start appearing when he will start suffering from immunodeficiency. Thereafter, the disease will start manifesting, and that may take a time period of maybe around 10 years. And suppose if the person has no knowledge, then meanwhile in this period of 1 to 10 years, he may infect a number of people. So the incubation period incubation period with respect to viral infection is very important. So what are the steps which are required to establish infection? So we have already discussed that in order to establish an infection, the virus must penetrate the epithelial barriers like skin, gastrointestinal lining, and respiratory lining. Apart from that, I think most of you have observed कि कभी अगर हमें लूज मोशन होता है, whenever we people suffer from loose motion, so in a general practice, doctors prescribe metronidazole, tinadazole, or maybe if it is very severe, then doctors prescribe norfloxacin or ciprofloxacin. I mean, this is a common observation which you all know. And in addition to this, you people have find that in addition to this antibiotics or antiprotozoal, doctors also number of times they prescribe certain tablets which are composed of lactobacillus spores. Or yeah, in case of home remedy, we people it's a very common experience that we people say, okay, if one is having a loose motion, then one should take banana and dahi, curd, because whenever you say curd is nothing but it's a in the curd that we have millions of spores of lactobacillus. So you people might be knowing that, for example, in case of human beings, we cannot produce vitamin D by ourselves. Inside our body, human beings don't have the ability to synthesize vitamin D. Inside our body, we have got lactobacillus inside the gut, and this lactobacillus are responsible for the production of vitamin D. So what I want to say is that inside the gut, inside our small intestine, and large intestine, you'll find that we have got normal flora, and in order to cause disease, these viruses have to compete with this normal flora for binding site. So in the what are those normal flora? Those normal floras are lactobacillus. Then we have got bifidobacterium. And nowadays you might be seeing that there is a increased trend of using probiotics. Now even you will find that even Amul ice creams comes loaded with probiotics. So, so in order to cause disease, these viruses have to compete with normal viruses. So even in case of females, the female genital tract, inside the even female genital tract, you'll find there is abundance of lactobacillus. So there always a war is going on between good microflora and a microflora and a flora which is going to cause disease. So if the ratio of normal flora is more as compared to those are going to cause disease, they will remain healthy. And suppose if those disease causing flora, if disease causing organisms, the number of those disease causing organisms it supersedes this normal flora, then a person becomes disease. So in order to establish an infection, the virus needs to compete with normal flora for finding sites with this lactobacillus or bifidobacterium, or we have got other probiotics also. Okay, now apart from that, inside our body we have got antigen presenting cells. So I will discuss it in the coming slides. So these viruses, in order to establish infection, they need to exert the innate immunity, which is comprised by macrophages, or in short we write it as M5. Then we have got neutrophils. Then we have got natural killer cells. So in order to establish infection, virus needs to exert there is a need to exert the innate immunity. So coming to innate or non-specific immunity, innate or non-specific immunity is comprises of anatomic and physiological barriers like intact skin, mucous membrane, temperature, acidity of gastric juices. There are certain protein factors, complement factors are there. Then we have got phagocytic barriers, we have got inflammatory barriers and fever. Then we have got mucociliary clearance. Apart from this, the innate immunity cells express certain TLR. So TLR stands for toll-like receptor. So, toll-like receptor is basically a family of different toll-like receptors, and their main function is to recognize viral signatures or bacterial signatures. Remember, everybody has got a signature. Like अगर हम लोग अगर अपनी बात करें तो everybody has got a distinct pattern of walking. Some of us कुछ लोग की ऐसी आदत होती है कि जब वो चलते हैं तो they makes a lot of noise by their slippers. So they makes a lot of noise. तो सबका अपना एक पैटर्न होता है and by the pattern you can say okay that person is this person is that. So similarly जो वायरस होते हैं जो बैक्टीरिया होते हैं हर वायरस और बैक्टीरिया का अपना एक सिग्नेचर होता है. So in terms of immunology, we call that signature as PAMP. So those signatures of the virus are known as pathogen associated molecular patterns. So activation of toll-like receptor signaling through recognition of PAMP, pathogen associated molecular patterns, which leads to the activation of transcriptional activation of certain genes which are responsible for coding, encoding pro-inflammatory cytokines, and we have got chemokines and what certain co-stimulatory molecules. And basically, in case of viral infection, the members of toll-like receptor family which participates are TLR3, TLR7, and TLR9. And in this also you will find that TLR3 is responsible for recognizing double-stranded RNA, TLR7 is responsible for single-stranded RNA. Remember, in our body, generally we don't have a double-stranded RNA, we have single-stranded RNA. So definitely if some double-stranded RNA is being detected in our body, that means it is something that is foreign, that might have come from certain viruses. So toll-like receptor 3 is responsible for the detection of double-stranded RNA. TLR7 is responsible for detection of single-stranded RNA. So this activation of toll-like family, toll-like receptors in response to pathogen-associated molecular patterns lead to the activation of this certain inflammatory cytokines, chemokines, and co-stimulatory molecules. So whenever we say cytokines, in the previous lecture also of Dr. Shweta, you people were listening about cytokines, and in today's context of COVID-19, आप लोग हमेशा सुनते होंगे cytokine storm. आप लोग हमेशा सुनते होंगे जो पेशेंट्स एडमिट होते बिफोर द डॉक्टर स्टार्ट ट्रीटमेंट और while treatment is going on, cytokine storm took place. उसके बाद क्या हुआ कि जो पर्सन्स के ब्लड वेसल्स में है ब्लड में क्लॉटिंग हो गई एंड उसके बाद देयर वाज उसके बाद आप देखोगे सीक्वेंसली आइदर देयर वाज अ फेलियर हार्ट उसके बाद किडनी फेलियर हुई हार्ट फेलियर हुई ब्रेन डेड एंड द पर्सन डाइड. So basically what do we mean by cytokines? Whenever we say cytokines, what are cytokines? So cytokines, so if you go through literature, you'll find that for cytokines from at a different point of time, sometimes people used to call them lymphokines, people used to call them monokines. Okay, so as understanding, as research progress, as understanding progress, all these lymphokines, monokines, chemokines, they were all grouped together under one heading that was cytokines. So basically, cytokines are low molecular weight proteins, and the most important thing about them is that they regulate the nature, intensity, and duration of the immune response by exerting a variety of effects on lymphocytes and other cells. Remember, this intensity and duration is very important. The major problem that we are facing during COVID-19 treatment is the intensity. If we'll go through when I will complete my class, you people might when I complete my talk, then you people might ask me, sir, if cytokines are very good, then why then people are dying out by cytokine storm? So the problem with cytokine storm is with respect to intensity. When we call about duration means a simple analogy I will give you. मेहमान अच्छा वही होता है जो हमारे घर आए अपना पर्पस पूरा करे एंड फिर वो वापस चला जाए। अगर मेहमान आपके घर में आके बहुत देर तक रह जाएगा तो फिर एक कंडीशन आती है जब आप लोग कहते हो अतिथि कब जाओगे। तो ड्यूरेशन इज ऑल्सो इम्पोर्टेंट। इफ साइटोकाइंस हैव बीन एक्टिवेटेड, so nature, intensity, and the duration or the time period for which it will act is very important. If the duration will be prolonged, rather than causing some benefit, it may cause certain harm. So cytokines are secreted low molecular weight proteins that regulate the nature, intensity, and duration of the immune response by exerting a variety of effects on lymphocytes and other cells. And when we say about cytokines, when we say cytokines include, we have got different types of interleukins like interleukin 1, 2, 3. Until date, there are more than 50 cytokines which are known in case of human beings. So we have got different types of cytokines. IL stands for interleukins. Until date, more than 50 different types of interleukins are known. Then cytokines include different types of interferons like interferon alpha, interferon beta, interferon gamma. Then we have got chemonecrosis factor alpha, chemonecrosis factor beta. Then we have got transforming growth factor beta 1, okay, beta 2. So the whole family of TGF. And in addition to that, we have got macrophage colony stimulating factor, granulocyte colony stimulating factor, GM, granulocyte macrophage colony stimulating factor. एंड मे बी इफ सम ऑफ यू, अगर किसी को अगर किसी ने किसी को कभी किसी के इन जान पहचान में किसी घर पे अगर कोई कैंसर पेशेंट रहा हो, भगवान न करे, तो उस केस में अगर आप देखोगे कभी अगर केमोथेरेपी चलती है, अगर हम कभी केमोथेरेपी, तो एक मेजर जो सीवियर साइड इफेक्ट होता है केमोथेरेपी का कि केमोथेरेपी लीड्स टू अ ड्रास्टिक रिडक्शन ऑफ प्लेटलेट्स, ब्लड प्लेटलेट्स। एंड जो ब्लड प्लेटलेट्स होते हैं उनको अगर नॉर्मल टाइम पे अगर उनको डिवाइड को प्रोलिफरेट करने का टाइम दिया जाए, सो it will take a large number of days. So what is the function of colony stimulating factor, CSF? Colony stimulating factor, within a very short time it can increase the colony. So in certain cases, number of times this CSF are administered. Anyway, so what I was talking about is just interleukins, interferons, TNF, TGF, and these different types of CSF, they all comes under the heading cytokines. So they all are cytokines. In addition to that, we have also chemokines. So why I am talking about cytokines, why it is very important. So it's a general experience that most of us, अगर कभी हम लोग को before this COVID came, before coronavirus, because before coronavirus and after coronavirus, the scenario has totally changed. कोरोना वायरस के पहले हम लोग जनरली वायरल इन्फेक्शन के लिए मेडिसिन सच नहीं लेते थे। अगर हम लोग डॉक्टर के पास भी जाते थे एंड जैसे कभी अगर कॉमन कोल्ड होता था या तबीयत खराब होती थी, तो डॉक्टर सर्टेन ब्लड टेस्ट करते थे एंड देन दे यूज्ड टू असेस। अगर उनको लगता था कि बैक्टीरियल इन्फेक्शन है, then in that case they used to prescribe certain antibiotics like ciprofloxacin, ofloxacin, amoxicillin and all, along with antipyretic like paracetamol, acetaminophen, and along with an anti-allergic like ceftriaxone. So that was true in case of bacterial infection. But कभी कभी डॉक्टर्स कहते थे कि ओके देयर इज नो नीड ऑफ मेडिसिन, क्योंकि आपको बैक्टीरियल इन्फेक्शन है ही नहीं, आपको वायरल इन्फेक्शन है, आपको तो कुछ नहीं लेना है, यू जस्ट टेक अ रेस्ट फॉर 5 to 7 days। हाँ, अगर कभी फीवर आता है तो यू टेक अ टैबलेट और पैरासीटामोल। ब्रैकेट में उनके प्रिस्क्रिप्शन पे एक टर्म डॉक्टर्स लोग लिखते थे, दैट वाज SOS। So if you are having fever, then take the paracetamol tablet, fever will reverse down, and maybe after 7 to 8 days or a week or 10 days आप अपने आप ऑटोमेटिकली ठीक हो जाओगे। तो क्वेश्चन इज कि क्या हम लोग सचमुच अपने आप ठीक हो जाते हैं, और देयर इज समथिंग दैट गोज इनसाइड आवर बॉडी। तो जब भी वायरल इन्फेक्शन होता है, अपने आप कोई ठीक नहीं होता है, हमारी बॉडी में इम्यून सिस्टम है, यू हैव गॉट। तो इम्यून सिस्टम में देयर आर डिफरेंट कंपोनेंट्स ऑफ इम्यून सिस्टम जो पार्टिसिपेट करते हैं एंड वायरस का हमारी बॉडी से वायरस को न्यूट्रलाइज करते हैं, वायरस को हमारे बॉडी से क्लियर करते हैं, एंड उसके बाद फिर हम लोग आफ्टर अ वीक और 10 days हम लोग परफेक्टली फाइन हो जाते हैं, irrespective of the fact whether you have taken medicine or irrespective of the fact whether you have not taken medicine. So this is something that I am talking about pre-COVID scenario. पोस्ट कोविड की बात अलग है। कोरोना वायरस आने के बाद जो वायरस के साथ नोशन था कि हाँ अपने आप ठीक हो जाएगा, दैट हैज that no longer holds true if anybody is getting infected by COVID-19. तो तो अगर आप इस ग्राफ को केयरफुली आप देखोगे तो ऑन द एक्स एक्सिस यू विल फाइंड दैट वी आर denoting time after viral infection in terms of days, day 1, day 2, day 3, day 4, day 5, 6, 7, 8, 9. And और आप लोग यहाँ पे you can see that there is a solid filled curve of yellow color का आप एक curve देख रहे होगे, that is denoting the virus titer. So virus titer inside the blood of a human person who is being infected by a virus. So जो yellow color का जो filled curve देख रहे हो, that stands for virus titer. So what we can see from this curve is that uh, as soon as a person gets infected by a virus, so follow this yellow curve. So during the initial phase, you will find that there is a steep increase in the titer of the virus. There is a steep increase in the titer of the virus from day 0 till day 1. After day 1, you will find that more or less this level of virus is no more, it's not growing exponentially. Rather, now it is assuming a stationary phase. So you will see that after day 1, the virus is assuming a stationary phase. Or maybe after 5, you will find that this virus titer has started declining. So the question is why? Why this exponential growth of virus has come to a halt? It has come to a stationary phase, and thereafter it's declining. What is happening inside the human body? What are the immune system? How come the immune system is encountering this virus infection? So what you will observe is that as soon as as soon as the virus infects a host, it starts growing its population, and as the population of the virus is growing, you follow this yellow filled box, yellow filled curve of the virus titer. So as the population of the virus grows, you will find that this red color curve stands for cytokines. This red color curve. So you'll find that as the population of the virus increases, the the concentration of the cytokines also starts increasing just a little after few hours. So you'll find that after, so to be precise, it's after a virus has infected a human body, human cell. 2 hours after a virus has infected a human cell, 2 hours after the infection, the concentration of interferons starts increasing. So interferons means the concentration of cytokines starts increasing. And you will find that this concentration of cytokines starts increasing and it peaks around day, in between day 1 and 2, maybe around day 1.5. And thereafter, the concentration of this cytokines, interferon alpha, interferon 12, interleukin 12, TNF alpha, it starts declining. At the same time, after some time, you will find that this blue curve represent the natural killer cell killing. So you will start that after on the just after few hours, of increase of cytokines, this natural killer cell population is also increasing. And something that I missed to tell you is that when you will find that when this cytokine has peaked, once the concentration or this level of cytokine has peaked around day 1 and 5, this exponential increase in the level of virus has come to a halt. Now it has come to a stationary phase. Vis-a-vis this increase of concentration of cytokines, there is an increase in the population of natural killer cells. And you will find that after cytokines, the level of natural killer cells population is increasing, and this natural killer cell population is peaking around day 3 and 4. Thereafter, this natural killer cell population starts declining, and around day 2.5, you'll find that this green curve, which stands for T cell killing, this T cell population starts growing from day 2.5, and it grows, and you'll find that as the natural killer cell population is increasing, and as T cell population starts increasing, and this pink curve, which is denoting the antibody concentration, you'll find that the concentration of antibody starts increasing around day 4. So as the concentration of T cell and the concentration of antibody starts decreasing, you will find that the level of virus, when we say virus titer means the concentration of virus in the serum of a human person. So you'll find that the concentration of virus titer starts declining, and as the concentration, as the population of T cell and the concentration of antibody increases, you'll find that around day 7, day 8, and 9, the level of virus has started declining, and to it almost comes to the base at around 8 to 9 days, and thereafter we become healthy and fine. So what? So what is happening? So you what in the initial phase, what you find is that it is the cytokines like TNF alpha, interleukin 12, interferon alpha, and natural killer cells that combine to provide early defense against virus infection. So we'll discuss that in detail. So immune response against a virus is primarily mediated through interferons. The name is interferon. The name interferon वर्ड है, अगर आप उसको स्प्लिट करोगे तो interferon means to interfere. अगर आप interferon वर्ड को स्प्लिट करेंगे, अगर हम लोग interferon वर्ड को स्प्लिट करेंगे तो interferon का मतलब होता है to interfere. वो किसके साथ interfere करेगा? It will interfere with viral replication. It will. So interferons का main purpose क्या होता है? To interfere with viral replication and thereby to create an antiviral state, both within the cell as well as in its neighboring environment. So interferon is a family which has got a different type 1, type 2. So we'll not get into the detail of those. So type 1 interferon production typically starts within the first 2 hours after virus infection. So irrespective of virus, जब भी किसी ह्यूमन या किसी भी ह्यूमन बीइंग के किसी होस्ट सेल्स के अंदर में जब वायरस इन्फेक्शन होता है, so within the first 2 hours, as you can see from the previous diagram, that within after this infection, as virus population increasing, after 2 hours, you'll find that the type 1 interferon production starts. Okay. And viruses and और जो interferon का production होता है, start क्यों होता है? Viruses like double stranded RNA, they induce the production of interferons like interferon alpha, interferon beta, interferon gamma, and this interferons in turn will produce an antiviral state by inhibiting viral replication as well as viral infection. And now the question is कि what are the cells which produce this interferons? जब हम लोग immune cells की बात करेंगे, which are the immune cells which are responsible for the production of interferon? So main producers of interferon alpha and interferon beta are plasmacytoid dendritic cells, which are also known as PDCs. And these are innate immune cells that circulate in the blood and are found in the peripheral lymphoid organs, and they constitute 0.4% of the peripheral blood mononuclear cells. So whenever you say the peripheral blood mononuclear cells, basically they stand for rounded cells. They stand for a cell with rounded nucleus, and that includes plasmacytoid dendritic cells, that includes lymphocytes and all. Okay. So if we'll go through this diagram, तो अगर आप यहाँ पे डायग्राम में देखोगे, we are having two cells. One is infected cell and the another one is plasmacytoid dendritic cell. तो यहाँ पे हम लोग देखेंगे, for example, एक particular cell है which has been infected by a virus. So in case of infected cell, you'll find these are the viral nucleic acid which are being recognized by RIG1. So RIG1 stands for retionic acid inducible gene 1, like this appears. So when cells become infected, RIG1 it detects the presence of viral nucleic acid. When we say viral nucleic acid, it will be either it can be single stranded RNA or it will double stranded RNA in the cytoplasm, and thereafter this RIG1 stimulates IRF3 and nuclear factor kappa B. So IRF3 stands for interferon regulatory factor 3. So after the activation of interferon regulatory factor 3 and nuclear factor kappa B, they are translocated inside the nucleus, and once they are translocated inside the nucleus, they will activate the transcription of type 1 interferon. So they will increase the expression of type 1 interferons that will lead to the production of this inflammatory cytokines or interferons. Similarly, in case of plasmacytoid dendritic cells, you'll find that plasmacytoid cells are having TLR. तो अभी हम जैसे previous slide में बताए थे, TLR stands for toll-like receptor 3, 7, and 9. So we are having different types of toll-like receptors. toll-like receptor families are different in case of bacteria. So certain toll-like receptor 379 is specific for viral infection. Certain other toll-like receptors are specific for bacterial infection. So toll-like receptor family receptors TLR3, TLR7, or TRL9, they recognize these viral nucleic acids, and thereafter they activate IRF7. So IRF7 means interferon regulatory factor 7, and once IRF7 is activated, it induces the production of interferons and inflammatory cytokines, and this once there is a production of inflammatory cytokines and interferons, so they act, which act on both the cell producing it. So they will. So you can see from the diagram, these interferons are being produced by plasma cytogenetic cells. So this plasma cytotic dendritic cells, this interferon being produced by plasma cytotic dendritic cells can act on the infected cell and at the same time also on the neighboring cells to establish antiviral state to resist viral infection. Okay. So there are certain signaling pathways which are responsible for the which are responsible by which the interferon induce the antiviral state. So what are those pathways? So interferon induce the antiviral state via there are number of pathways. I will talk about two. One of them is 2' 5' oligoadenylate synthetase pathway. The another one is synthesis of protein kinase receptor, is a PKR pathway, and the third one is ribonuclease L. So the first pathway that we're talking about is oligoadenylate 2' 5' oligoadenylate synthesis pathway. So here we will find that so upon ligand binding. So ligand means suppose if we want to charge our for undergraduate students, what do you mean by ligand and receptor? तो अगर आप देखोगे अगर आपको अपनी मोबाइल को अगर चार्ज करनी है तो आपके मोबाइल में चार्ज करने के लिए आपके पास एक अडेप्टर होता है and adapters you have got two pins. So in order to charge आप अपने अडेप्टर को कहाँ फिक्स करते हो? I'm giving this example in a very crude way. तो आप क्या करते हो, जो सॉकेट होता है सॉकेट में आप प्लग को लगाते हो एंड जैसे तो आप इसको इस तरह का इक्विवेलेंट मानो, दिस प्लग इज इक्विवेलेंट टू लाइगेंड, और जब वो सॉकेट में बाइंड करता है दैट इज इक्विवेलेंट टू रिसेप्टर. तो जब लाइगेंड रिसेप्टर से बाइंड करता है तो फिर क्या होता है, वायर के थ्रू करंट फ्लो करने लगता है. ओके, तो सिमिलरली जब भी हम लोग अगर अपने किसी भी इम्यून सिस्टम या फिजियोलॉजिकल पैरामीटर की बात करेंगे तो वहाँ पे क्या होता है, जो लाइगेंड्स होते हैं वो रिसेप्टर से बाइंड करते हैं एंड आफ्टर द लाइगेंड बाइंड विद द रिसेप्टर दे एक्टिवेट अ पर्टिकुलर सिग्नलिंग पाथवे. तो यहाँ भी आप देखोगे, फॉर एग्जांपल इन केस ऑफ दिस वायरस इन्फेक्टेड सेल limphide, that's interferon alpha or interferon beta. This interferon alpha and beta is acting like a ligand which is binding with the interferon alpha and beta receptors. And once this interferon alpha and beta will bind with interferon alpha and beta receptors, it will induce the expression of oligoadenylate synthetase. It will induce the expression of oligoadenylate synthetase. Once there is increase in the expression of oligoadenylate synthetase, तो यहाँ पे आप देखोगे पहले क्या था, RNase L. So for undergraduate students, whenever we say RNase, DNase, proteinase, what does that mean? RNase means something which can degrade RNA. DNase means something which can degrade DNA. When we say proteinase, something which can degrade protein. Okay. तो यहाँ पे आप देखोगे जो RNase है, initially the RNase was inactive. Previously we are having inactive RNase L. So once there is increase in the expression of oligoadenylate synthetase, this inactive RNase L is being converted into active RNase. And once this inactive RNase L becomes active RNase L, this will result into the degradation of what? Degradation of viral RNA. So once the virus RNA is degraded, it will lead to the inhibition of protein synthesis machinery. Thereafter, the replication or increase in the number of viruses will stop. So this is one mechanism by which interferon signaling induces the antiviral state. The second pathway is synthesis of protein kinase. Okay. So what happens during synthesis of protein kinase? तो अगर आप लोग इस डायग्राम को केयरफुली देखोगे तो यहाँ पे दो तरह की चीजें हैं. Remember, यहाँ a war is going on. हम लोग जहाँ पे भी बैठे हुए हैं, अगर आप लोग जहाँ पे भी बैठे हो, the surfaces that we are touching, the environment in which we are breathing, it's full of bacteria and virus. The war is constantly going on. The question here is कि कौन जीत रहा है? So, if you are healthy, that means our immune system is able to take care of all these bacteria and viruses which is present in our environment. Okay. And अगर कभी हमें डिजीज हो जाती है तो दैट मीन्स उस पर्टिकुलर मोमेंट के लिए हमारा इम्यून सिस्टम कहीं ना कहीं पे फेल हुआ है. तो अ कंटिन्यूअस वॉर इज गोइंग ऑन. हमारी जो इम्यून सिस्टम है वो हमेशा बैक्टीरिया एंड वायरस को न्यूट्रलाइज करने के लिए काम कर रही है. एट द सेम टाइम this bacteria and viruses, they are devising strategy, they are devising mechanism so as to how to cause disease. So in this slide I will give you an idea of both how these viruses are trying to devise mechanism to overcome our immune response, and at the same time how our immune system is trying to overcome these viruses by activating certain mechanism. Now, so you will find तो अभी क्वेश्चन ये था कि जो इंटरफेरॉन्स है वो how they create an antiviral state via this pathway. So the name of this pathway is protein kinase receptor. Now, so in the diagram you will find that cytokines and states they activate the this PKR. Once after activation you will find there is यहाँ पे एक और step हो रहा है, that's a dimerization step. You will find there are dimers of PKR is forming, and at the same time you will find यहाँ पे क्या है EIF2 alpha. EIF2 alpha stands for eukaryotic initiation factor 2 alpha. तो जैसे पीकेआर का डायमरेशन हो रहा है, एट द सेम टाइम वन मोर इवेंट इज टेकिंग प्लेस, दैट इज phosphorylation of EIF2 alpha. So you'll find that as soon as dimerization of PKR takes place, there is a phosphorylation event. EIF2 alpha, that is a eukaryotic inhibition factor 2 alpha, is getting phosphorylated. And once this EIF2 alpha gets phosphorylated, there will be inhibition of translation of. Okay. So at the same time, this after a dimerization of PKA, some other mechanism can also participate, like it will lead to the activation of IQAC gamma pathway, which will lead to the activation of nuclear factor kappa B, and this nuclear factor kappa B can lead to the activation of antiviral genes. Okay. Apart from that, it may also activate to some extent JNK pathway. We have got a number of pathways inside our body system, or it may also activate P32, P38 MAP kinase pathway. Okay. Or it can, or suppose if it finds that okay, it's not possible to remove the virus, so it will kill the virus infected cell by activating the apoptotic machinery. Like it will activate FADD, then it will activate after which there will be activation of phosphatases, and thereafter there will be apoptosis of the virally infected cells. So activation of double-stranded RNA dependent protein kinase receptor disrupts virus infection by phosphorylating and inhibiting eukaryotic initiation factor EIF2 alpha, and hence blocking the translation of. So here you have seen that once there is phosphorylation of EIF2 alpha, it leads to the inhibition of translation. So hence blocking the translation of viral mRNA, and by inhibiting apoptosis and pro-apoptic BCL2 cascade dependent mechanism killing cells before virus can be released. So it will kill the virus infected cells before the virus is released into circulation. Okay, so this is one mechanism. As I said, a war is going on. जैसे हमारा इम्यून सिस्टम वायरस को न्यूट्रलाइज करने के लिए इट इज डिवाइजिंग सर्टेन मशीनरी. Similarly, viruses in order to survive, viruses in order to pass on their progeny, viruses in order to cause infection, they have also adopted certain mechanisms. For example, you will see यहाँ पे जैसे आप देखोगे certain viruses like यहाँ पे आप देखोगे रियो वायरस, इन्फ्लुएंजा ए बी, SCMV, वैक्सीनिया वायरस, ये सारे वायरसेस क्या करते हैं, दे ब्लॉक दिस स्टेप. कौन से स्टेप, दिस डायमरेशन स्टेप ऑफ PKR. Or there are certain viruses like vaccinia virus or swine pox virus. What they do is, in order to survive, what they do is they prevent this, they block this step. Which step? This phosphorylation step of EIF2 alpha. Similarly, you will find there are certain other viruses like HSP1, rotavirus. They inhibit this particular step. So these are the different steps by which these viruses, by blocking those steps, they are trying to ensure their survival and their propagation.
Excuse me, Rajkumar, I would just like to interrupt you. Actually, we have also got one more speaker. If you just
Sorry.
a few minutes more.
I will.
Sorry that I had to tell you this.
How many minutes I have got, Angana?
No, actually your time is up. It's already 4:10. Okay, so
I will try to wind up within 5 minutes.
Okay. Thank you, Dada. Thank you.
Okay. So there is one more mechanism, interference-induced antiviral state by another mechanism, that's ribonuclease. There is where deamination of adenosine takes place into inosine, and thereafter there is activation of RNA cell, which leads to a degradation of RNA. Okay. So this is how interferons lead to the arrest the replication of viruses. So after that, no, after that the next player that comes into scene is natural killer cells. So just I'm giving an idea that on the surfaces of different cells you find there are certain receptors like T cells express certain receptors like CD2, CD4, CD8, BD8. So natural killer cells they are identified by their expression of CD56. So this interferon signaling, what interferons? So once interferon signaling, once interferons are activated, what they do is they signal, they activate natural killer cells, which starts eliminating virally infected cells. So these natural killer cells are activated by interferon alpha, interferon beta. Natural killer cell, and then thereafter what they do is these natural killer cells they target and kill the virus infected cells. Okay. So going to this slide, you will find this is a mechanism. This slide depicts the mechanism by which these natural killer cells act. Suppose this is a normal cell and this is an abnormal cell. So what is the difference between these normal and abnormal cells? On the surface of the normal cell, you will find there is a ubiquitous molecule, there is an MSC class 1 molecule, and this natural killer cell have got two receptors. One is a killer activating receptors, one is a killer inhibitory receptors. So this, in case of normal cell, you will find that this MSC class 1 molecule will fill this killer inhibitory receptors. So there will be no attack. But in case of abnormal cells, they are lacking this MSC class 1 molecules. Since MSC class 1 molecules are absent, these natural killer cells will initiate the reaction of kill. They will start producing perforations and enzymes, and by this complement pathway, they will kill an abnormal cells. That means a virus infected cell. Okay. So after this, you'll after the action of interferons and natural killer cells, you'll find the next player that comes into scene is that they are the T cell followed by antibody. So there are cells which are known as antigen presenting cells, and antigen presenting cells include macrophages, B cells, and dendritic cells. And what does this antigen presenting cells do? तो अगर आप यहाँ पे देखोगे चाहे वो वायरसेस हो फंजाई हो यानी बैक्टीरिया हो pathogens are being eaten by these macrophages or dendritic cells, and thereafter this they are known as antigen presenting cells. So जो macrophages होते हैं या dendritic cells होते हैं, there are two pathways, endogenous pathway and exogenous pathway, where those pathways they process these pathogens, and thereafter they display portion of the antigen on the surface. So you can see in this diagram, this is the foreign antigens which are being displayed on the surface of these macrophages. And once these foreign antigens are being displayed on the surface of the macrophages, helper T cell will detect them and it will release cytokines. So once helper T cell is activated, at the same time helper T cell can activate cytotoxic T cell or it can activate T cells. If cytotoxic T cell is activated, cytotoxic T cell name itself is cytotoxic. That means it is going to kill the infected cell. At the same time, it will lead to the formation of certain memory T cells, which will participate. If suppose in future, if again the host cell encounters that particular virus, and this activated T cell can also activate the B cells. And once B cells are activated, B cells mature into plasma cells, and once B cells mature into plasma cells, they start producing antibodies. At the same time, they will start producing. At the same time, we have memory B cells, and this memory B cells they form the principle of vaccination. Okay, so activated helper T cell reduce the production of interferons. And I'm skipping certain slides now. So cytotoxic T lymphocyte has been observed to peak at around 7 to 10 days post-infection, and therefore they eliminate the virally question is how come antibodies protect against viruses? तो अगर आप इस ग्राफ में देखोगे तो सबसे पहले cytokines followed by natural killer cells they come into scene, that is followed by T cell killing, and thereafter T cell killing जो सबसे लास्ट में जो आता है that आता है antibodies. Even in case of COVID-19 you will be finding that जो अभी एंटीजन एंटीबॉडी इंटरेक्शन बेस पे जो किट आई है उसके भी हम लोग केस में कहते हैं कि दैट इज किट दैट यू कैन यूज फॉर वेदर अ पर्सन वाज initially that's not for whether the person is suffering from COVID-19 or not. If a person has a previous exposure to COVID-19, that kit is used for its detection or to ascertaining whether a person was infected by COVID-19 or not. Okay. So how does antibodies protect against viruses? Antibodies bind to viral surface antigens and they are efficient in preventing attachment. So virus entry in the cell is mediated through certain receptors. So try to understand. जो virus होता है in order to show its antigenicity, that virus needs to bind to the receptor. And antibodies क्या करते हैं? Suppose if antibody is present, what antibody will do is it will surround the viruses. Once the antibody will surround the virus, this virus will not be able to bind to its corresponding receptor. And if this virus will not be able to bind to the receptor, this virus will not be internalized into the cell. If the virus will not be internalized into the cell, the virus genomic material, it will not be able to bind its genetic material into the host cell genome, and thereafter replication process will be halted. So virus entry into the cell is mediated through certain receptors, and you will find that in case of certain viruses like influenza virus and rhinoviruses, they bind so just for example, influenza virus binds via sialic acid, rhinovirus binds through ICM. So if receptor is blocked, infection is blocked. Okay. So this is how antibodies produce. They inhibit the viral replication. Okay. This is my current research team, and this is my current area of research. These two students have recently been awarded their PhD thesis. These two students are currently working. Thank you. Thank you, Angana.
Thank you, Dr. Koiri, for your
exceeding the time.
No, it's perfectly okay. We can actually understand that while talking about a subject we love, we actually tend to forget our time. So it's absolutely okay for you. Okay, now I would like to go on with the technical session, and I would like to thank Dr. Koiri for your edifying and instructive speech, which would make us aware about the coronavirus infection. Now, another announcement which I already made, that please post your comments in the comment section of the YouTube channel so that the questions can be taken up. Now, our next speaker is Dr. Momita Shamonto. I feel extremely blissful to get her as one of our resource persons. She has graduated from Bethune College, Kolkata, and has acquired her MSc degree in organic chemistry from Calcutta University. She has obtained her PhD in protein chemistry, molecular biophysics from Indian Institute of Science, Bangalore, under the able guidance of Professor P. Balaram. She pursued postdoctoral research and gained expertise in different technical spheres from the Baylor College of Medicine, Houston, US, and University of Pennsylvania, Philadelphia, USA. She has published different articles and scientific papers in highly reputed international journals. Formerly, Dr. Shamonto headed the biotechnology department at the Centurion University, Bhubaneswar, Orissa. She is currently working as a senior scientist at Dr. Reddy's Laboratories, Hyderabad. She is a determined and well-rounded scientist with strong analytical skills, thorough understanding of biosimilar drug development in industry, with effective interpersonal and writing skills. Dr. Shamonto's work involves different chromatographic techniques, spectroscopic methods, and mass spectrometry for the therapeutic monoclonal antibody. At last but not the least, after her illustrious achievements, I could not resist to share with our viewers that she is my friend and a very humble, down-to-earth person. Today, Dr. Shamunto will share her ideas on the present scenario of COVID-19 pandemic and would also talk about the therapeutics and health aspects. Please may I have the pride privilege to hand over this forum to Dr. Shamunto for her talk.
Thank you, Angona, for your very kind words. I'm really, really humbled. And thanks to all of you for providing me this opportunity to present in front of you all. So just to give a bit, Angana already said a lot about me. Just to say that initially I was in academics. So in 2017, I transitioned to industry in the biopharmaceutical sector. So today's talk, actually, I will be trying to give you that insight from an industry perspective at where we are and how we are doing with respect to coronavirus. So I'll go ahead and share my screen. So can you see the screen, all of you?
Yes, the screen is being visible. That's okay.
Okay. Yeah. Yes. So actually, just in the line of what Dr. Sumita said, it's a bit uncomfortable, like giving a talk like this without really seeing the faces of all the audience. But anyway, this is the pandemic situation, and I'll go ahead. So the title of my talk is 2020, the year of war with COVID-19. So this is the kind of outline of my talk. So for the first few slides, I'll really go quick because it is just to set up the context of the talk with respect to the vaccines that I'll be talking about for coronavirus. So the first point is the current picture. Then what are we actually up against? Where are we now? And then the final point, when is it going to be over? Well, that is $1 million question, but I'll try to give a perspective from industry aspect. Going to the current picture. So you all are aware of this current picture. So this is the update from yesterday actually. So global cases total, it is 32 million right now, and total number of deaths globally is about 1 million. So you can see the clusters, the red ones, it is really bad. We all know the numbers actually. US is really heading, then we have India, followed by Brazil, Russia. And you can see in the figure, the red dots are really clustered around the eastern parts of North America. There are some in Southern America, in India, and we all really know from news that few months back, it was really bad in Italy, Spain, Germany, and England. So coming to India, this is the current situation. The numbers are really going high day by day, and currently it is about 58 lakhs, and the total number of deaths are very close to 1 lakh. So what are we up against actually? So what is the cause and what is the result in this corona? So I know that we all, the previous two talks, we had been talking about how it started, about the virus, and that really actually built up a very good platform for me to continue going ahead. But I'll just briefly touch up these as a point. So the beginning, as we all know, it's started from a probable bat to human infection. It started with a bat being the host, and there were a lot of arguments and confusions about whether it directly came from the bat to the human, or there was an intermediate animal which was involved in the transition. So the earliest known person with symptoms was later discovered to have fallen ill on 1st of December 2019. So that is ideally the first time it was detected and this corona came into picture. So what happens actually when corona invades our body? So as very beautifully described in the previous talks, it has this proteins on the surface, and one of the protein is the spike protein, or the S, and that gives a crown-like impression, and therefore it derives its name corona. And this S attaches to the human ACE receptor protein, ACE2 receptor protein, and that helps or allows the virus to enter into the human cell. So the virus basically then hijacks the human cell and then it starts replicating its own viral DNA. More and more viruses are churned out inside our body, and the overworked cells basically died after that. Again, about the cytokine storm. So what happens? Okay, most patients recover, so that is the good news. But the conditions of severely ill patients, they become more severely ill because of some irreversible damage to vital organs. So because of this, our body goes into this SOS call. It sometimes gets confusing, and therefore that triggers a poor immune response. And sometimes it is followed by cytokine storm. So that is a do or die phase, suicidal mode, where actually a very uncontrolled overexpression of immune molecules are triggered, and that causes the damage to several organs, and eventually basically the body shuts down. Again, a quick review. The total numbers are 32 million, active are 7.5 million, and total number of deceased are around 1 million. No specific drug yet, there's no vaccine yet, and there is no proof of herd immunity yet. So the way we are doing is maintaining physical distancing, wearing masks, and maintaining proper personal hygiene, which seems to minimize the risk to some extent. So when is it going to be over? So this is a very tough question which none of us have an answer for. But we can say that when it might be over, because we all are looking towards the vaccine. The vaccine seems, one is the cure and other is the vaccine. So that we all are looking towards the vaccine. So which are the giant biopharmaceutical companies that are in the process of making vaccines? So not only biopharmaceutical industries, the academic institutes are also involved in making the vaccines. So I'll just give you a brief description or an update on how it works. Because in newspapers you see these are the front runners, these are the vaccines that are coming up, these are the vaccine candidates, these have made to the third phase clinical trials, these have got approval, this vaccine has got licensed. So sometimes it really gets confusing, because I want to give you an idea about how much time it actually takes for a vaccine when it comes out of a laboratory to the bed side of a patient, or it gets injected into the patient. So that really consists of several stages, and it is not a very quick thing, and it doesn't have a really quick turnaround time that it can be done really quickly. So I'll give you an idea about the conventional timelines and what happens when the world is facing a pandemic, if that process can be made really fast. So vaccines, we all know, it creates immunity in an individual by introducing the weakened or the killed form of the pathogen. It may be bacteria, or it may be virus, or the toxins, or any of the surface proteins. So it induces the acquired immunity, so that when the actual disease causing agents tends to intrude your body is ready to mount a defense against it. So that is what a vaccine is. I'll briefly now take you back to the history of vaccine. So these are some of the cases against which we have vaccines, though there are many other also diseases. But here I have tried to summarize a few. So you will know the red point is when actually the agent that was linked to the disease or it was recognized, and the green point at the end is actually when the vaccine was approved or registered and it came to the market. So you can see, you can have an idea about the timeline, how long it usually takes for a vaccine to come into the market. It of course has several depending factors on which it depends. Like on first is how long it takes to understand the agents that cause it. Maybe it can be the protein structure, the virus information about the virus, and then the work starts in the lab, and all the exploratory work is done in the lab. And once that is finished, it goes through several stages of clinical trials, and then once those data is approved by the different regulatory bodies, it gets approved, and finally it is ready to be launched into the market. So you can see from here actually I have added for Ebola, the virus, because this is one of the recent vaccines that came up. It got approved in December 2019, so it should be in market. And for among the rest, you can see for measles, it took 10 years. So these are the quickest turnaround times which have happened in the history of vaccination innovation. So now going to what actually happens in a vaccine research development. So the top panel which you see in this slide is actually the traditional timeline, which you can see it is 10 to 15 years. So this exploratory part which is on the left is the one which takes place entirely on the bench in your laboratory. So that is when we investigate the structure of the different proteins, the viruses, the sequence, the DNA sequence, the RNA sequence, so whatever scientific investigation is needed is done in the exploratory stage. And then we develop the vaccine also in the lab bench and do it at the lab scale. So once that exploratory phase is successful, your vaccine is, or the vaccine candidate is ready for the next few stages. So the next stage which comes after the exploratory stage is the preclinical stage. So that is absolutely mandatory when you do the trial of the vaccine in animal models. Well, that can be in the lab as well, but it is done in animal models, and once that is successful, you move on to the clinical trials. So the clinical trials are divided into several stages: phase 1, phase 2, phase 3. You can see from the number of human structures that can give you an idea that phase 1 is usually done with a very low number of subjects, and the number of subjects gradually increases as we move from phase 1 to phase 2 to phase 3. And then once each of these stages are successful, then all the data and everything is reviewed from the clinical trials by the several regulatory boards, and they are, if they are okay, then they are approved. So I have written FDA here, which applies to US, because that is the approval body of drugs, of any new drugs or vaccines in US. But usually if any vaccine or drugs are developed, they usually go for FDA for approval, because usually if a drug or vaccine is FDA approved, it can be allowed in the other countries, unless some country has very strict requirements with respect to its own regulatory board. So I'll just say that the analogous, the board in India is called CDSCO. So once that approval is ready, approval is received, actually the drug can be manufactured, and the manufacturing sites also get the approval, and the drugs can be manufactured in bulk, and then it can be in the market and the patients can access that. So that is a very traditional timeline. Now, in case of corona, we really cannot afford 10 to 15 years when it is a global pandemic. So what happens in such cases? The timeline is really accelerated very aggressively, as you can say, and as you can see here, and it is a situation which is called emergency use authorizations. So all these regulatory boards and approval boards, they all work in collaboration, and they start seeing the data from really early stage, and therefore the stages are overlapped, so that it can really be condensed over a smaller time, and so that the patients can really access it much early. So that is what is happening with respect to coronavirus. Now we are really looking for a year or maybe 18 months maximum to have a vaccine in the market. Let me tell you, it's not that whatever candidates start from exploratory stage make it to the manufacturing stage. It's only 15% of vaccine development have a success rate, and they really make it up to the patient. The others really get halted at different stages because of some issues or problems. So now I'll discuss each of these phases a bit in detail. So the first is preclinical stage. So once you have the vaccine ready, and to gain that confidence to inject into a human subject, it is done in animals. So the most common animal models are mice and monkeys, because monkey is the most related non-human primate. And once these are done, so in these study, what actually is done, the safety of the vaccine is of the prime importance, which is evaluated here, how safe the vaccine is, and then whether it is able to provoke the immune response, it is able to generate the antibody. And these studies give researchers the idea of the cellular response which they might expect in the humans. And also they get an idea about the safe starting dose for the next phase, so that they can safely administer it into a human subject. So many candidate vaccines, as I said, they never progress beyond this stage because they fail to produce the desired immune response. It usually takes 1 to 2 years. Now coming to the phase 1 clinical trial. So this is the first stage when a vaccine is injected into a human subject. So at this point in phase 1, all the tests are done on healthy subjects or healthy volunteers, and the number varies from 20 to 50. And there is usually no placebo group tried at this stage. I will describe what is placebo group. So what happens when a vaccine or a new drug is tried in a clinical trial? So there are two groups. One is a placebo group, where actually you do not have the molecule, the actual molecule. You have everything, the background formulation and everything except the molecule. So that is called the placebo molecule. And the other is your real vaccine. So the way it is done is patients, not patients at this point, sorry, the healthy subjects are randomly assigned whether an actual vaccine or a placebo one. And then so that the purpose is to get unbiased results. So the safety of the candidate vaccine is assessed at this point. Then the type and the extent of immune response that the vaccine provokes, that is studied. And also dose escalation is done at this point. So what is meant by dose escalation? The dose is gradually increased to know at what particular dose we see an effect, or the antibody starts getting generated, or to what particular dose level side effects starts appearing. So they do this kind of study at the phase 1. So once we get through the phase 1 study successfully, we move on to the phase 2. So phase 2 involves a bigger group of volunteers. It is about 80 to 100. And here actually trials are done often with a placebo group. The efficacy of the vaccine is assessed here to know how well the vaccine works. The safety is again assessed here, but here it is being assessed over a more bigger population and diverse population. And test dosage and immunization schedules are like conclusions are made on these at this phase. Again, when the phase 2 is successfully done, we move on to the third phase. Now, third phase is a very, very pivotal study. As you all know, and you will be reading in newspaper and even in the news and all, like how the different coronaviruses are at what particular phases it is in the clinical trials. Like you all have heard about Sputnik V and several others, like Serum Institute is coming up with a vaccine which is from the Oxford University, which phase it is in. So we all are reading about these different phases. So what happens in this phase 3? So this is really a pivotal study, but it is the extremely important part of the clinical trial of a vaccine. It includes the highest number of volunteers. It is about 100 to 1,000. And these are randomized trial along with a placebo group. It verifies and confirms the safety of the drug in a typical patient. So here the volunteers are mostly patients. Here we are not talking about healthy volunteers. And this is a larger population. It verifies the efficacy of the vaccine, how well it works. So all the confirmations of these parameters like efficacy, safety, dosing are confirmed at this stage because it is being done with a much bigger population and a diverse population. So therefore this is very important. It is very important for a vaccine to give productive result after this phase, because only then it can be applied for a license, and it can be registered as a vaccine. So now I'm going to phase 4. So ideally it involves 3 phases, and after these 3 phases it is evaluated, approved by the different regulatory bodies, and then it is launched into the market. The phase 4 happens parallelly actually. The approval does not depend on the phase 4, or it doesn't have to wait for phase 4. So this study is conducted post the vaccine launch, and while it is in the market, it is evaluated on many more subjects, like more than thousands. It can be 5,000 to 10,000. So it confirms the safety over a longer period of time, because by now we have been evaluating from phase 1 itself the safety, and phase 3 also goes on for several years, and therefore phase 4 gives more confidence because it has data on the safety of the vaccine for over a longer time. Sometimes, because it is done over a huge population or much larger population, the rare adverse side effects of a drug are like observed at this point, at phase 4 study of a drug. So those are observed and those are monitored. Also, another outcome of phase 4 is when we do new dosages, new evaluation of doses or the duration of the treatment. So those are sometimes re-evaluated at the phase 4 test of a drug. Now, the question is, what happens after clinical trials? So say if a vaccine candidate has successfully passed all those stage gates, like phase 1, phase 2, phase 3, what happens after these clinical trials? So it is usually a new drug application that is done to FDA or whatever the governing body it may apply to in a country to approve that new drug, and eventually followed by sale and marketing. So all the compiled data is evaluated, which has been obtained from the preclinical stages all through to the clinical stages. So here what actually is observed is how safe the drug is, how effective the drug is, and what are the benefits. Like, does the benefits outweigh the risk? So is the drug's proposed labeling appropriate? So once all these assessments are done and FDA gives a nod, then the vaccine is ready to be launched into the market. Now, launching also, there are several other parameters that the biopharma industry really have to gear up before the launch. So, one of that very important thing is supply. So, supply is very connected to the demand. So, you can imagine in what stage the world is right now and the way all are looking forward to a vaccine. So, that means it should be able to cater to a huge number of population, able to supply that vaccine. So that means they should have that manufacturing sites ready to that level, or to produce that vaccine to that scale, to cater to the masses. So now, coming up to the corona vaccine race. So actually we are in this context, right? Corona vaccine, it is really a race against time, because we really do not have that 10-15 years to develop a vaccine, and we really have to make sure that we have the vaccine ready within a few months, at the same time maintain the quality and the effectiveness of the drug. So we all have heard about this Sputnik V vaccine, right? I just found few of these really funny and pulled them out from the web, because it's like, who is the first one to give a vaccine now? So, going back to immune response again. I know this has been discussed a couple of times now. I'll just do once really quick, because my following slides will be on what are the ways we can make vaccine, and that is connected somehow to this one. So, coronavirus, as already said, it has the spike protein on the surface. So, the spike protein, or the S, which is on the outer surface, it actually locks to this ACE2 receptor on the surface of the human cells. And these cells translate to help in the invading the virus into the human cells. And once inside, the cells translate the virus's DNA to produce more and more viruses. And that is how it multiplies within our body. So what is the immune response? So the immune response is, we have this antigen presenting cells, APC, which recognizes this virus, and they trigger or activate the T helper cells, the B cells, which in turn generate the antibodies that mask this virus and prevent, and also the cytotoxic T cells, which destroys this virus containing cells. And in the meantime, we have this long-lived memory B cells and T cells that keep on patrolling the body and maintain the immune system, so that they are ready to attack when we again have this pathogen invading our body. So based on this, there are 8 different approaches on which corona vaccine is being made right now in different parts of the world. So these 8 approaches I have tried to summarize here. One is the live attenuated virus, which is actually the weakened virus that can be used as a vaccine. The other is the weakened virus, which can be used as a vaccine. The 3 and 4 are where we can use a DNA material is used in the virus. The 4 is, I'll talk each one of these in detail in the following slides. This is just an overview. The 4th one is when we can use the RNA material as the virus. So each one of these get into the target cells, and in some way they trigger the immune response. The 5th and 6th are when we can use the viral vector. It can be replicating or it can be non-replicating. And then 7 and 8 are when we can use the protein of the virus to develop a vaccine. So VLP is virus-like particle. They are basically antivirus. And the H1 is subunit spike protein on the surface which can be used as a virus. So these are the 8 approaches or 8 platforms on which people are making vaccines right now. So the first one is the virus vaccine, where the actual virus is used as a vaccine. So the first kind is weakened virus or attenuated virus. This is a live virus. It is weakened by picking up a mutation that is less probable to cause a disease. However, it induces strong, long-lasting T-cell, B-cell immune response. It is inexpensive and it can be produced really rapidly. So the current vaccines that are in use are BCG, smallpox, MMR, chickenpox, rotavirus, yellow fever, and polio. Inactivated virus. This virus is made infectious, non-infectious actually, by using chemicals such as formaldehyde or heat. And currently the vaccines that are in use are polio, HIV, rabies, hepatitis A, flu. Now currently both approaches are being used to develop coronavirus. So I have written the viruses which have really made it to the clinical phases in red. So weakened virus approach is also being used to make a corona vaccine, but probably it's still in the development phase. But you can see what is highlighted here in red, that vaccine, that COVID-19 vaccine candidate, the Picovac by the Sinovac Biotech. So that has taken up this approach of inactivated virus, and it is in the clinical trial phase. You should be seeing it shortly. So going ahead to the next approach is the viral vector vaccine. So here again, the first type is, it has two types. One is a replicating viral vector. So here viral vector vaccine replicates within the cell, and that's how it promotes a strong immune response. Currently, the vaccines that are used are hepatitis B, pertussis, pneumonia, HPV, HIV. Another kind is the non-replicating viral vector. So here, actually currently no licensed vaccine is in use which has used this approach or this platform. So actually you can say it is really a very innovating approach. Though it has been long used for gene therapy, but no vaccine registered or approved vaccine is in use in this approach. But however, we all have heard about the University of Oxford, the vaccine that is being developed by University of Oxford, right? So that is the COVID vaccine candidate which is being developed on this platform of non-replicating viral vector. Actually, the Sputnik vaccine, which has recently been in use in Russia, that is also based on this approach, non-replicating viral vector. So what that vaccine has done, it has taken up a adenoviral vector as a means of like transferring part of the COVID-19 material into it, and that's how the vaccine has been made. So that has also taken this non-replicating viral vector. And I'll give you a kind of landscape of how we are doing with respect to the different approaches to the vaccines that we are making with respect to distribution, what I mean, across these 8 approaches. And you would notice that actually the major vaccines are being made on this approach of non-replicating viral vector. Okay, the second one, not the second one actually, I think this is the 5th approach. It is the nucleic acid vaccine, where actually the genetic material used, not entirely. So the first one is DNA vaccine, where they use the DNA, but only a part of the DNA that corresponds to the coronavirus protein. And here electroporation method is needed to make pores in the membrane so that it can initiate the uptake of this DNA into the cell. And the RNA vaccine, where the RNA is actually enclosed into a lipid envelope so that it helps in the proper transportation of this RNA into the cell. And then once it goes into the cell, it translates into the respective proteins, viral proteins, and then it triggers the immune response. So it is also unproven in practice so far, and the candidate COVID-19 vaccine, DNA vaccine, is one by Inovio Pharma, Korean Institute of Health, and the mRNA approach is being taken by one of the giant pharmaceuticals company, Pfizer. So these are all again in the clinical trial stages, and by clinical trial I really mean by the later stages, that is stage 3. So after that, the nucleic acid vaccine, these are the other approaches, which is protein-based vaccine. So protein subunit. So in this there are two types. One is the protein subunit, where basically you make the virus's spike protein. So if you have the virus's spike protein in your vaccine, then it is recognized and it triggers the immune response in the same way. So hepatitis B, it was the first subunit vaccine made, and influenza vaccine. So those are currently now. And corona vaccine candidate, which is one of the very promising ones, upcoming ones, is by Novavax, which is based on this technique. The next technique is virus-like particles. So it is also called VLP. So what happens? It is actually a antivirus shell. So it doesn't have the genetic material inside. It just has the envelope and all the surface proteins on it. So it is non-infectious because it doesn't have the genetic material, but it triggers a very strong immune response. But it has a flip side of it is very difficult to manufacture. I personally have worked on virus-like particles, VLP of norovirus in one of my postdoc projects, and I knew how difficult it is to make this intact VLPs at a lab scale. So you can imagine making a vaccine where one of the very important thing is your method really have to be robust so that it can survive the scale up. So that is why it is very difficult to manufacture, keeping the intact structure of the virus-like particle. So one of the examples is human papilloma virus vaccine. So though corona vaccines are also being tried in this approach, but none of them have yet made to the clinical stages. Okay. So this is the landscape for coronavirus in the current scenario. So on the top left, you can see how the vaccine developers are working on it day and night. So it is like the relative distribution. Certain percentage is by academia, certain percentage is by industry, and certain percentage is the partnership between several institutes and industry. On the top right is the different approaches, the different 8 approaches that I discussed right now, and how currently these viruses are being developed. Please ignore that human papilloma virus vaccine. I think it was a copy and paste error from some other slides. It actually applies for COVID-19 vaccine trials. Okay. So you can see here, as I was discussing in one of the previous slides, so you can see here subunit is one of the major approaches, and the viral vector is also one of the major approaches, or one of the major platforms that has been taken up to make the virus, to make the vaccine. So currently the distribution of how these are at different stages So in the lower panel, DNA, RNA approach, subunit, inactivated, and non-replicating viral. So you can see that the red part, the non-replicating part, is really high in this bar chart in the below panel. So mostly the viruses that the vaccines that are making successfully are going ahead to the several stages clinical trials. They are mostly being made on this non-replicating viral strategy. Some are definitely on the DNA and RNA strategy, and also some are on the inactivated virus strategy. So this data I have taken from Nature Materials, and I have put the reference here. There is also a very interesting tracker, the COVID-19 tracker by milkeninstitute.org. You people really can go into this site at any time, and it is a tracker for all vaccines. So whatever vaccines are being made in the world for corona, and whatever stages they are, what are the approaches, and I mean everything is there in that site. So I really found this site helpful, and I thought I'll bring it up here. So yeah, this is the landscape of for coronavirus right now. In the next slide, I will just discuss like what are the front runners right now. So we know that several biopharma industries are working on making this vaccine, and some have really started at the development stage, some have made to the preclinical stage, and some have made to this clinical stages. So some of the front runners, as you can see, are from Sinovac, Sinopharm, Oxford, AstraZeneca, Moderna. I think Moderna was one of the very first biopharma companies to initiate making this vaccine for corona. I mean, one of the very first one, because it was in news in the very beginning of April, March and April at that point of time. Then there are some more. Pfizer started a little late, but it is really catching up. Inovio, Novavax, Johnson & Johnson, and Merck. So you would hear that a lot, as I said, you could hear that a lot of those companies have started making vaccines, but it's really a few which really meet or finally reach the end line, because it's not very easy to pass through each of these phases. It also depends on the quality of the vaccine, how safe they are. And once they reach those endpoint, then it gets authorized. And right now for corona vaccine, it is an emergency authorization, and it gets approved, and finally it gets launched in the market. So these are some of the candidates, like in an overview I wanted to bring forward, like which we can expect vaccines in the next few months, if not by end of this year, probably in the beginning of next year. Out of these, we have heard a lot about this Oxford vaccine, and the Serum Institute is clubbing with them. Yeah, mostly these. And Sputnik V has already, it has, it has also been in news, and Dr. Reddy's have gone into an agreement with Russia to assist in the clinical 3 trial study in India, and also cater to the need of whatever number of vials of this vaccine in India. So I hope it was helpful. This is like I think it was a bit different than the other talk in a way, because I wanted to bring forward this current scenario of where and how of the vaccines, and where we stand, and how we can look forward to with respect to vaccine. So from this, 141 vaccines are currently in the development, and in human trials is 25. So this is a month-old data. So probably the vaccines in human trials will also probably be increasing a bit more. And in development platform also, I think some more candidates have also stepped in by now. So that is all from my end, and I hope together we shall overcome. And other than vaccine, if we keep and maintain, trying to maintain the safety norms that we all know, we will able to control to some extent. But of course, finally, the vaccine would be the answer. But we will really have to wait till they come on the shelf. Thank you all.
Thank you, Dr. Shamanto, for your insightful, informative talk and making us aware of the possibilities that would instigate a new ray of hope in the near future. We are immensely illuminated. With this, we have reached the flag end of today's technical session. So now I would request Dr. Tapan Kumar Pal and Dr. Shreyoshi Gupta, Department of Zoology, to continue with the question hour forum.
A very good afternoon to all the dignitaries and our audience, all the speakers also. We get a large number of questions in our YouTube pool. We have selected few of them, and my first question, the first question asked by Silpi Sarkar. What are the normal symptoms for pandemic attack during COVID, and what are the hormonal changes related to pandemic attack COVID? To Sumedha, madam.
Hello, Shilpi.
Silpi, what are the normal symptoms for pandemic attack, and what are the?
Are you talking about panic attack? I think it is panic attack.
Panic attack, right? Panic attack, right? And what are the consequences in hormonal changes?
Yeah, there are many hormones that actually take part in this panic attack initiation, but the initial symptoms, you cannot generalize the thing, because one can have an anxiety problem, one person can have the panic problem more than the other. But the normal symptoms is breathlessness, sweating, and the heart starts beating like anything. It seems that you are tired, you are drained out, like that. And there are many hormones that actually participate in this panic attack, and in which I remember one neuropeptide, which is hypothalamic neuropeptide, produced from the hypothalamus, and its high level is actually related to panic attack, and it is known as orexin. It regulates the wakefulness of a person and appetite of a person. So these things are going to get affected when a person gets panic attack. And there is also a stress hormone that is very common. Maybe all of us know about adrenaline. And sometimes people having thyroid hormone problems also means hyperthyroidism is also means hyperthyroid like symptoms are also associated with stress response sometimes. So I think anything more that can be added by the other speakers or anyone in the platform, that will be very good. But now these things are just coming to my mind. So I hope Shilpi can understand that.
Thank you, ma'am. Rajkumar, yes.
There is one hormone that is known as cortisol. So whenever there is a stress or panic, the level of cortisol naturally increases. Even under a normal condition also, suppose if our students are under stress due to exam, even in that condition also level of cortisol increases. So be it any factor, be it exam or be it due to corona pandemic, the level of cortisol is expected to increase. Okay, thank you.
Next question asked by Shraddhanjali Das. How much brain damage could occur due to COVID-19? To Dr. Sumedha Roy.
That is very difficult to say it right now because time and again in my lecture I have told that it is only 10 months of COVID-19. God forbid that it should not be extended, or else we come to know about it. But best of luck to the scientists who are fighting for the vaccines. But yes, there can be massive impact in the brain if the COVID infection persists in the body and in the latent condition of the virus, because we all know that virus can remain latent. Many viruses can remain latent in the body, and we don't know about COVID-19 that I shared in my slides, but about some relatives of COVID, SARS-CoV, other one that I have mentioned, that maintains a latency. So if it is latent in the brain, then the cells are going to get damaged, because in severe COVID infections, it has been demonstrated already that in patients there is severe damage in the cells. There are necrotic patches. There are some of the images that you may remember from my slides where there are signs of damage in the brain.
Okay, thank you. Next question asked by Pooja Ahuja. She asked, what is the possibility of the recurrence of the second recurrence attack of the COVID-19, and what is the lifespan of the antibody if generated in the first attack? By Pooja Ahuja.
This question is directed to which speaker?
Pooja Ahuja asked a question to you, Somedha Roy. The question is, what is the possibility of the recurrence of the pandemic attack, COVID-19, whether it generated of the antibody, and what is the lifespan of the antibody?
Yeah, that is very difficult to say. Means there are varied different opinions that are coming up across the globe, because some people who have had infections, they don't show up the antibodies after 14 days or 21 days, whereas now a recent data has come up that people are seen to have these antibody levels, because when in Europe, in the first phase of infection, it was seen that after 15 days, some people don't who have had infections in the COVID-19 infections and have been detected positive, they don't show up the antibodies after 15 to 21 days. It was seen that at that time, but now there are some studies which are coming up that show the persistence of the antibody still. So I might ask help from Dr. Shamanto regarding this, about the persistence of the naturally developed antibody which against the COVID-19 infection.
Yeah, Madam Momita Samanto. Yeah. The question was actually asked by Pooja Ahuja during the session of the Sumedha Roy. That's why I'm asking you. But actually, question related to Dr. Momita Samanto. You are
Yes, right. Can you please repeat the question?
I'm
actually internet connection is.
Yeah, I'm repeating the question. The question is, what is the possibility of the recurrence of the COVID-19 attack, and what is the lifespan of the antibody that are produced in the first attack?
So from you all can hear, right? So actually, see, the antibodies generated, I mean, for all of us who are in the science platform, have the science platform, we know that once the antibodies are generated in the body, they usually have this lifeline, lifelong they can survive to fight that pathogen, right? But for COVID-19, there has been literature where it has been seen that COVID infected patients, like after 3 months, they have got infected again. So again, yeah, so this is again a window, I think, where in which a lot of scientific research is going on. I cannot really pin down as number of months, but I can say definitely, but it is not like the other conventional infections. Say for chickenpox, we have antibody, and we acquire a lifelong immunity. It is not like that. But for how many months and why is it happening? A lot of research is being done in that, and we have observed this. So there has been cases where maybe the antibody has degraded over 3 months or 4 months.
Okay, thank you.
But yeah, I mean, I hope that answers to some extent.
Yeah, you are quite okay. Thank you. Thank you, madam. Okay. Our next question asked by our faculty member, Dr. Angana Ghoshal. How does the cytokine storm triggered by SARS-CoV-2 contribute to the neurological impact of COVID-19? To Dr. Subedhara.
Yeah. As I have mentioned in my lecture, I think, Angona, it is clear that cytokine storm is actually pro-inflammatory. So it initiates some inflammatory signal in the tissue. So since we have the ACE receptors, ACE type 2 receptors in the brain and the neurons and the glial cells, so there can be association, or the virus can directly invade the neurons. There is probability of the virus getting entry into the neurons or the glial cells, and thus initiate the pro-inflammatory response. And if it initiates, means there is the normal cytokine response, and when there is excess of cytokine produced in the neuronal tissues, then there is if the excess of the cytokines will actually cause the pro-inflammatory response in the neuronal tissues. And this pro-inflammatory response or pro-inflammatory signal is quite similar to that which occurs in other neurodegenerative diseases. So that is where the scientists are a little bit curious. That is where the scientists are speculative that these pro-inflammatory response initiated by the cytokine storm may direct the neurological tissue towards degradation and degeneration,
Okay.
like other neurodegenerative diseases.
Right. Thank you. Our next question asked by Prerana, Miss Prerana. The question is: after recovering COVID-19, what are the chances to be get infected by other diseases? To Dr. Rajkumar Poedi.
Could you please repeat the question again?
What are the chances to be infected by other disease after recovering after COVID-19?
If I remember well, recently there was a literature which suggests that persons who have been infected with COVID and have recovered, so it has been observed in around 20 to 30% of cases that persons who have recovered from COVID-19, now they are suffering from diabetes. Initially they were healthy, they were not suffering from diabetes. Still the mechanism is not known. But after having recovered from COVID-19, now they are suffering from diabetes. So as far as COVID-19 is concerned, it's causing certain other diseases. Maybe the person is recovering from COVID, but they are suffering from certain other diseases. So one report that is associated with diabetes is available in internet. So still more findings are yet to come, because just people have just started working on COVID-19, and it's hardly a matter of 6 months. So still we don't have much information on that. Maybe Dr. Samantha and Sumedha may add to something.
Yeah, I must just say that people who have had already the infection of COVID-19, I have the opportunity to talk to some of them. The thing that they are really missing is the energy. So they are very tired. They feel very tired. They feel very drained out after recovery also. When they have resumed their office, they are doing their job, they have come back to the office, still they don't have the same amount of energy that they used to have. So that is the first thing that I have noticed in these patients. They are no more patients now because they have recovered from the infection, but still they don't feel that energy, and they don't feel maybe that zeal. It is also a trauma because after this being quarantined and being locked down at home, they had suffered some mental stress as well. So these are the things that need to be taken care of after the patient recovers from the typical viral infection.
Madam, one question: is the change in the sleeping behavior after COVID?
After COVID, there is a chance of change in sleep. After COVID, I don't know, but during this COVID related panic or anxiety or stress, there is definitely a change in the sleeping behavior. And means it is very obvious that a person who is having a regular job, regular security in that person doesn't have any problem related to sleep, because he has had enough of work done in the whole day. But when a person is quarantined and is locked out in one small room and is suffering from the anxiety of uh, there is another anxiety very much related to our day-to-day life, that is the tension of losing the job, the tension of financial crisis, the tension of having less commodities available after I recover. So if so these things this phase of pandemic has affected the sleeping behavior not only of the patients but also the people who are locked out or locked down at home, uh, away from the family, away from the near and dear ones. So it is another very important to be in touch with others, at least through the virtual platform, and to talk to others. We should also be compassionate, because another point is very important that I actually want to share, maybe, that sometimes people who have infections, uh, they are not treated the way they were treated previously. Now we want to just keep away from them. That is another thing which actually causes mental means a pain in those patients. It's not only from the virus that they are suffering, it is also the social virus which is also affecting their mental status. So that is another important thing that we should take care of, and sleeping behavior in that case will obviously be affected.
Okay, thank you, thank you.
And Tapan, if you are going to believe that report, that persons who have recovered from COVID are showing diabetes-like condition now. So you can yourself imagine that if a person suffers from diabetes, so with the passage of time, the person suffering from diabetes have neurological complication, they suffer from some kidney failure, they suffer from certain heart-related problems. So if such report is to be believed, that a good number of persons now are developing diabetes after recovering from COVID, then these are the diabetes associated complications that may arise in near future.
Okay, okay. Our next question to Dr.
Yeah, I mean, in line to Dr. Rajkumar actually and Dr. Shumeda, it's basically our body is putting up a big fight, the immune system. And since the immune system is off guard, our body is more susceptible to different infections and diseases. So that is basically the thing. And that's what comes to, like, we are feeling weak, and again the mental part is already there. So these two things are working together and having a cumulative effect.
Okay, okay. Thank you. Our next question to Dr. Koiri. If a person having dengue, and what are the chances having dengue, recovered from dengue, just recovered from dengue, what are the chances to be get infected by COVID-19? Is there higher immune compromised stage
So,
of the person?
look, chance
Question
is
asked by
fairly high. And suppose if
Pin-
because dengue and COVID, even if a person is recovering from dengue, that means his immune system is already very weak, and now if he suppose he has an exposure of COVID, the chances of infection taking place is very high.
I think the question was actually whether the platelet count may be compromised in that case. But question not the thing. I think platelet counting downward during the viral infection. The question regarding that one.
Tapan, then I would like to add one thing. Still, I don't know how much that report is authentic. Recently, while going through literature, I came across certain papers which were saying that aspirin, I mean the aspirin that we take, dispirin or aspirin, salicylic acid, that can be used as an antiviral agent. And the mechanism they had suggested was that aspirin acts as a blood thinner, and it has been observed in the past also that aspirin has got certain antiviral activity. But it's still a matter of research whether this aspirin can have antiviral activity against COVID-19 or not. And in this, and basically they were, when they were talking about aspirin, basically they were focusing on this protein factor only from vaccine-related activities, whether the blood platelets are they are aggregating or not. So there is some relation. Still, there is some relation. If you suppose if the concerned student is interested to know, he may do a little bit of literature survey on aspirin, how aspirin can be used to for treatment of COVID-19, and maybe a correlation may exist. Still, I don't remember everything in the paper. I just had a superficial overview. But there are reports with respect to aspirin. There are certain scientists who are suggesting that aspirin can be a drug of choice for the treatment of COVID-19. But that still remains to be validated. And the mechanism has got to do with blood clotting factors.
Okay, thank you. I request Sreyoshi Gupta, Dr. Sreyoshi Gupta of our department, to continue the question session, and I hand over the podium to Dr. Sreyoshi Gupta. Mute. Your mic is mute. Please unmute, Dr. Gupta. Okay.
So thank you, sir. Basically, first of all, I want to thank you all for all your patience, and directly going to the questions for Momita ma'am. The question is from Nivedita Kormokar. She has asked about the mutation and virus and the challenge of vaccine development. Can you please comment on something about this? Thank you.
Sorry, can you repeat the first part? The virus, some virus?
Yeah,
or
virus mutation and the challenge for vaccine development.
Yes, I know it is in the context of Corona. I'm sure we all of us have read that part, right? Because Corona is like a highly mutating virus, and I'm sure, I mean, we all know that scientists have been working on or investigating this aspect as well. And if you see the geographical distribution as well, there are the viruses which are probably in Latin America or in US or in Europe might be slightly genetically different than the one in India, or maybe which might be different again from China. And it is not just with geographical location, it might be also with respect to time. So maybe a virus which was there maybe 6 months before at the same place, it might be a bit different after 6 months more. So yes, that is why again that question which was asked with respect to antibody, the previous question I asked, it's difficult because the antibody might not, one of the reasons could be that it is not able to work lifelong because the virus is also quickly modifying. Yes, that is a challenging aspect, but again, scientists, when they develop a vaccine, they really work on that aspect to see which particular target they want to pin down to develop the vaccine. So probably out of the entire structure, whether it is genetic material or they are working by the protein approach or whatever way, they want to pin down a particular unique part or a unique section which might not change over time, and that way they might develop the vaccine, and so that way it would make it robust. But yes, this is one of the challenges in developing a vaccine as well, I must say. Yeah, so that is also determines how much time is invested in the exploratory section of the virus development. Sometimes it becomes very challenging for the vaccine itself to get out of the lab because they are still working on that aspect. Yeah.
Thank you, ma'am. There is another question from my side. Like there is a buzz recently going on around all over the world that what is important, is it a nasal vaccine or is a muscular vaccine or it's intravenous vaccine? So what is your take on that? Is this nasal vaccine is going to be our savior and we are going to we can actually leave our old normal than that of this new normal? So what is your take in this? nasal vaccination and muscular vaccination or the intravenous vaccination? What is your? Thank you.
Yeah, so one of the parts of vaccine investigation or the science behind making vaccine is uh, the way it is given to the patient. Okay, so it may be muscular, nasal, and all those. And so one aspect is how the molecule can be kept stable. By molecule, I mean the main ingredient of the vaccine. So some of them can be really dried and stored, cold dried and stored. You must have seen several vaccines. Some are in liquid formulation, and that can be cold stored. So if they are cold stored, there are extra challenges about shipment, transporting over several countries. If the manufacturing site is say coming from US, and we have this huge demand, so there are several aspects which have to be considered while developing a vaccine. One is the storage, how stable the molecule can be kept safe, and another is the way it is given into the human, that convenience. So going by that, yeah, mostly these are will be through injections like muscular. Nasal vaccines, I am not very sure right now, but that might be also approach. I mean, different industries have their different approaches, and they verify all these points and make sure that they have a robust approach. Yeah.
Thank
Okay,
you,
ma'am.
ma'am.
Yeah, actually, I'm very sorry to interrupt the questioner session, but actually the service providers who are actually carrying on with this online broadcast, they have actually given me a time limit of 5:30. Okay, so although there are a lot of questions and lot of people are wanting to interact, I have to end today's technical session. But for our viewers, I would like to say that we will share our email ID of our department. If you wish to share your questions, you can always share it with our speakers. Okay, so I'm very sorry that I've got to end this session now. So at last, I would like to say that I extend my heartfelt gratitude and thanks to all our resource persons who have found time for us from their busy schedule to share their valuable thoughts with us and make this webinar a success. We would also like to thank Dr. Ashish Kumar De, Principal TDB College, the members of the IQAC and Seminar Committee, and all our faculty members for their unstinted support, without whose motivation this would not have been possible. So with this, I declare today's forum to end, hoping to meet you all tomorrow from 11 o'clock in the morning. And I hope that all the speakers will be able to join. Thank you.
Thank you.
Thank you.
Okay, thank you all.
Thank
You all.